Acosta Montella, Minister of Finance and Public Credit of the Republic of Nicaragua, and the Nicaraguan Russian Committee. Marta Reyes Álvarez, who is the Minister of Health of the Republic of Nicaragua. Roberto López Gómez, Executive President of the Nicaraguan Institute of Social Security. Alexander Kokolikov, Ambassador Extraordinary and Plenipotentiary of the Russian Federation in the Republic of Nicaragua. Victor Turkin, Director of the Center Institute of Vaccines and Serum. We would like to have the participation of our special guests who are with us today. We are privileged to have them here. Jose Francisco Javier Moore, who is the Ambassador of the Republic of Venezuela. Ambassador of Cuba. was created in the Washington Institute because it has helped the most vulnerable groups, children, elderly, and pregnant women, and those who have risky health situations. We have granted access to quality, free of charge vaccine to our people. We also have the cooperation and help of the Russian Federation through vaccines and also help from other countries like China and other agencies that have helped us acquire more vaccines to increase our vaccine coverage in the country, which is about 96% of the population has at least one dose of COVID-19 vaccine. And this sets us in the first place of the full scheme for full dosage in the Latin American region. Also, in the joint work, we have strengthened aspects that have to do with accompaniment by WHO and WHO to strengthen our national regulatory agency with advancements from 2020 to 2022, also the law on substitutes, the law creator of the sanitary regulations, regulation of good practices in transportation of medical supplies, and our surveillance, and the approval of the regulation for clinical trials of biological products in human beings, and the agreement with Cuba as a country to produce biological products, agreements between institutions that are also part of the functional regulatory system and the creation of the Department of Quality Assurance based on risk. This was created over the past 3 years to strengthen the development of our regulatory agency and to produce and to move forward in the creation of quality vaccines for the global market. We are still moving forward. We've made great progress, but we need to keep moving forward because we need to have a regional level regulatory agency. This is why we are working with other countries and our third government. For us, since 2007, we want to restore the right to health care for all of our citizens, free, equal, and provided for free to the population. Thank you very much. New challenges. Russia is still advocating for multilateralism and solidarity and the cooperation. There are no borders because there is a common threat and we need a common joint response in close collaboration and cooperation. It is unacceptable to politicize the reception US help. Russia highlights the urgency of equity and these coverage has facts speak for themselves. In 2021, our country became one of the major vaccine providers for the country. We provided over 200,000 full doses of Sputnik V and over 3 million doses of Sputnik. Russia is willing to also help by means of technology and is open to knowledge exchange in epidemiology, in healthcare. As since 2008, we have created a national education project called Biomika, which aims at raising awareness of new professions in the field based on achievements from the pharmaceutical production industry. This is a long-term project. Another example is the Russian cooperation with Latin America through the Biotechnology Institute Mexico that has been working in Nicaragua since 2016. Since 2019, the Institute started producing the first anti-flu vaccines that was used to vaccinate over 3 million Nicaraguans. Now we make shipments of the anti-flu vaccine to Cuba and Venezuela. The products are also registered in Guatemala, in El Salvador, and the Dominican Republic. As for other diseases, the Meshnikov Institute has started to work on the creation of a dengue vaccine. And we do all the work in cooperation with the government of Nicaragua. Currently, we are aware and we are alert of new diseases. The government of the Russian Federation donated to Nicaragua vaccines against smallpox. And this is to create or to have a reserve for when smallpox or monkeypox cases present themselves. Russian institutes are conducting research to find medications for the different diseases. This forum is also a tangible proof of the open policy of Russia. I would like to highlight that vaccines and the technology from Russia is a result of arduous work of our scientists which is now helping us combat quickly and swiftly the current challenges that we are living. Russia is willing to share these achievements through cooperation with other allies, and this is a proof of our firm commitment to sustainable development to all, and this is based on the principles of common good. I wish most success for this Congress. Thank you very much for your attention.
Para dar por concluida esta ceremonia inaugural,
To
con
conclude
ustedes las
this
palabras del
inaugural
compañero
session,
Víctor
we
Trujín,
will hear
director
from
del
Dr.
Instituto de
Victor
Vacunas
Trujín,
y Sueros de
the
San
director
Petersburgo
of
de
the St.
Rusia.
Petersburg Institute of Vaccines and Sera. Thank you.
Всем добрый день.
Good morning to all.
Уважаемые члены президиума, уважаемые послы
Dear
многих
members
братских стран,
of the
уважаемые присутствующие
Presidium,
специалисты и
ambassadors
гости
of
нашего
the different
еще не
countries
юбилейного,
that
но
are here
уже
present
седьмого
at
Международного
this
конгресса.
Congress. It's the 7th International
Огромный всем
Congress
привет
between Russia
и
and
пожелания
Nicaragua.
удачи в
I
работе
greet
от нашего
all of
министра
you from the
Вероники
bottom of my
Скворцовой.
heart and wish you
Хочу немного
success.
сказать о политике в области
In the name of
фармы
Minister
и здравоохранения,
Veronica
о темах
Skvortsova.
и
Let me
целях
just tell you
Конгресса
about a little bit
и
about our
о людях,
pharmaceutical
которые
policies
его
and explain
ведут,
also a
которые
little
в
bit
нем
the
участвуют.
aims of the
Последствия
Congress and the
потери
people who
от недавно
are
прошедшего
participating in it.
урагана в Никарагуа
The
составили более
consequence
400
of
миллионов долларов.
the recent
И
hurricane
это
in Nicaragua, for
природная
example,
катастрофа, с
led
которой
to more
бороться
than
весьма и
400
весьма трудно.
million
Но против понятных
dollars,
природных
and
проблем
it's a
народное
national
правительство успешно борется,
catastrophe. But there are other
осуществляя
issues that are also
серьезные расходы
natural,
на массовую
and
вакцинацию
the popular
населения,
government
и это
struggles
значимо
against them by
защищает и
providing
от эпидемий,
resources
и от пандемий,
via
и, возможно, еще
large-scale
больших потерь народного хозяйства.
vaccination of the
Сейчас
population against, for
Никарагуа
example,
расходует более
pandemics, diseases,
580 миллионов
and
долларов
trying to
на
avoid other losses. Today,
здравоохранение
Nicaragua
и
spends more
более
than
400 миллионов
$580
на образование.
million a year on health and more than $500 million on education.
Институт
The
вакцин
Mechnikov
Санкт-Петербурга
Institute
в этом
and
тренде
the
общего
St. Petersburg
взаимодействия.
Institute of
В этом
Vaccines and
году
Sera
количество
is
обучаемых у нас
following then this
студентов
tendency
еще
of
выросло.
cooperation.
Новый
The number of
студент
students
Альдо
that
Баланес
are now
сейчас
in
в Томске.
Russia
Томск
has grown.
северный город, но это самый
And now
академический город
we have one
в
student in
Сибири, в
Tomsk
России.
in the north, very
Спасибо
good
посольству
academics
России в
area
Никарагуа
in
и
Siberia. And we thank the
сотрудничеству
Russian
за
embassy in
устойчивую и
Nicaragua
многолетнюю
and
политику в этой
Rosatuzhnistov,
области.
who are implementing a sustainable long-term
Тут должно было
policy
быть
in
фото,
this
я
sector.
его не вижу.
Here,
вот оно.
there should be a picture. There we go.
Всегда в последнее время шучу, что я забрал 4 никарагуанцев
I
из
always
Никарагуа,
say
а
sort
вернул
of
5.
jokingly that I brought 4 Nicaraguans from Nicaragua and I'm giving back 5.
Благодарю Марту Рейс, нашего
And
министра здравоохранения, за
I am
блестящее
very
выступление
grateful to
на
Marta
форуме
Reyes, the
GMP в
Ministry
России,
of Health,
в моем родном городе
for,
Иркутске. Это тоже Сибирь.
Having worked with Irkutsk,
Кстати,
also in Siberia, which is where I
глава
was born,
Института
and
надлежащих практик
she has
Министерства
worked
промышленности и
closely
торговли России,
with
одного
people
из
in Kursk,
столпов
and also
российской
good
школы
practices
GMP,
in the industry
доктор
of commerce, which is one of the
Владислав Шестаков,
pillars of good
тоже
practices
здесь.
in
И у
Russia.
нас сегодня будет,
Dr.
наверное,
Vladislav
самая
Shestakov
интересная
is
сессия
also
по
here
регуляторной
today,
науке.
and I think that will be
Международная
one of the
регуляторная
most interesting
практика
section when we
во
talk
время
about
ковида
the science
шагнула
of
значительно вперед.
regulating
Это и новые типы и
medicines.
платформы
We
вакцин.
have learned a lot
Это
from
массовость и
COVID.
интеграция
We've seen
производств,
new
различных
platforms,
производств в
new
мире.
types of
Это
vaccines,
новые подходы в
large-scale
исследовании
integration of
регуляторной науки
types
за
of
компаниями,
vaccines in the
разработчиками
world.
и производителями.
Again,
Помните,
progress has
когда-то
been made
доктор
in
Рафаэль
the
Перес,
regulatory sector.
руководитель
I don't know if you remember,
регулятора
but
ЦЕКМЕД
when
на Кубе,
Rafael
очень
Perez,
грамотно и
the
очень
head of the
далеко
SECMED in
видящий
Cuba,
об этом говорил, что регуляторика должна
very
следовать за
well,
производителями, что
highly
производители и
educated
разработчики
and
теперь
far-seeing
всегда
person
будут
always
впереди.
said that
Сегодня с нами еще один мой друг,
the
главный
producers
эксперт
need to be at the
Минздрава
vanguard,
России
and the
по
regulations should
фармацевтическим
follow
препаратам
the
и в частности по вакцинам,
industry, the
доктор
Ministry of
Вадим
Health and
Меркулов.
Pharmacology
И нас
and
ждет
vaccines in prison.
еще
Dr.
одна
Vadim
самая
Merkulov,
дискуссионная
who at
лекция.
one point will be,
Конечно, самая
give us a
глубокая
class
благодарность
here. He
всем
really will no
регуляторам,
doubt generate many discussions.
принявшим приглашение
And so
на наш
I
конгресс,
thank
и всем,
all
кто смог
regulators
приехать в
for their
Никарагуа,
participation
ну, а также
as they
находится с нами
follow
на
the
удалении.
industry
Не
and
только
their
мы
presence here at
отмечаем,
this very
мир
important
давно
event in
стоит
Nicaragua, also
на
those who are
границе
online.
понимания, что регуляторные
And we
правила
have for a long
и
time have
требования
seen
– это тоже
that
наука, и
the
она тоже должна
world
меняться. то
is
есть
coming to
развиваться.
understand
Нам всем
that
чрезвычайно
the
важно развитие
regulation of
НРА
pharmacological
Никарагуа,
products is a
так как
science,
разработки
and it's
и
really
производство не
important for
стоят
all
на
of us
месте.
to
Скоро
develop
нам потребуется
these
сильнейший
regulatory
орган,
agencies
свой
and for the
регуляторный орган
design and
Никарагуа.
product of
Мы готовы
medicines.
написать план
And here in
выхода
Nicaragua
НРО
too,
Никарагуа
they
на
never stop.
уровень работы
And
с
now with
вакцинами.
Menschikoff here in
Мы
Nicaragua,
готовы
we will
привлечь
need to advance in that
международных и
as well.
независимых экспертов
And we
для
will be
полноценной работы
supporting
по
also
ключевым
the
аспектам
Nicaraguan
развития
regulating
НРА
agencies so they can work
Никарагуа.
with
Это
vaccines
управление качеством, это
and also
инспекторат,
a
это
plan
GXP,
for the
это
development
лицензирование.
of qualities,
Полагаю,
good
что
practice,
необходимо
and so on
как
in
можно
the
быстрее
field of
завершить
vaccines
проект
and
и
pharmacological products.
определить
And this is a
финансирование на
project we need to
создание
really move along
этого
with as quickly
независимого
as
и
possible
компетентного
and
НРО,
to really
который встанет
create
в один ряд с
this
ведущими регуляторами
agency and
региона,
make it competent, and
стран-производителей
it will
вакцин,
act then at
Мексики,
the level
Бразилии,
of other
Аргентины, Кубы.
similar
На регуляторной
organizations
сессии
in
будут
Cuba,
высказаны
Argentina,
позиции
Brazil,
ассоциации
Mexico,
DCVMN.
and at the same level.
Это
And we will
ассоциация
be hearing about
производителей
this during
вакцин
the Congress.
развивающихся стран, с
As
которой
well,
прибыли мои
we
коллеги
will
Анатолий
hearing also
Евтушенко
from developing
и
countries,
Сергей Аракелов.
for example, Anatoly
По
Евтушенко
Мечникову
and Sergei
хочу
Aniklov, and
поприветствовать присутствующую сегодня с
as
нами
concerns
Анну
Meshnikov,
Трешур,
I want to
представителя
greet Anna
Панамериканской
Treshul.
организации
He
в
is here with
Республике
us
Никарагуа.
today. The
Роль ПАОЗ в создании и
representative of the
становлении
Pan
Института
American Health
Мечников
Organization
сложно
in
переоценить.
Nicaragua.
С первых дней тогда
It is
еще проекта
impossible
трансфера технологий
to
производства
exaggerate
перекозной
the
вакцины
role
из
played,
России в
and
Никарагуа,
also the Mexican
страновой
Institute,
офис и
when
штаб-квартира
we were
ПАОЗ
first talking about the
оказывали
Russian
всестороннюю помощь и
anti-grip
поддержку. Директор
vaccine,
Панамериканской
and here in Nicaragua
организации
we got all the
Кариса
support
Этьен
necessary.
была на открытии Института
The
Мечников
director
в 2016 году.
of the Pan American Health Organization was at the
Выполнение
inauguration
всех
of the Mexican
производственных
Institute in
планов – это
2016.
уже прекрасная норма в развитии всей
With all
команды
the
Мечников.
production plans are
Им,
very good
команде
indicator of the
Мечников и
development of the
руководителю,
Menshnikov team.
директору
And
Института
the
Мечников
Menshnikov team
Станиславу
and the
Ульба
director of the Menshnikov
наш
Institute,
мой низкий
Stanislav
поклон
Ульба.
и большое спасибо
I
за
again very,
делаемую
very
работу.
grateful and great respect
У
for
Мечника
the work
также
that they have
стоит
been doing
задача
thus far.
поставки
Menshnikov
вакцин в
also
традиционные
has
страны и,
the
конечно,
task to
развитие
supply
на новые регионы
vaccines
Центральной
to
Америки
traditional
и
countries,
Мексики.
but also developing countries,
У
and
Мечника
now
еще
entering
одна
Central
интересная
America and
была
Mexico.
отработана модель и
Menshnikov has
позиция.
another
Это
interesting
достаточно
model,
жесткое, такое трудное
which is
клиническое исследование по требованию Всемирной организации здравоохранения,
clinical,
около
very
1000
difficult,
добровольцев,
complex
занявшее более
clinical
года,
study
но выполненное
with over
блестяще
1,000
совместно
volunteers
с
for over
Министерством
1 year,
здравоохранения,
but it was
совместно с
complied with
Институтом социального
100% with
страхования
help of the World
и
Health Organization
университетами.
and the
Большое
Ministry of
спасибо
Health,
доктору Елене Начаровой
Social
за
Security
эту
Institute and
фантастически
universities,
сложную
and
работу.
Elena Nacharva did a
Я видел ее
tremendous
фотографии,
job
где она
because
прямо
this
с
really
собакой
was
ночью в
a
Министерстве
challenge. I've
здравоохранения
seen pictures
сидела и
where she and her
готовила
dogs
материалы
are
для
at the
инспекции.
Ministry of Health at
ВОЗ.
night preparing materials
Также
for the
отмечу
World Health Organization.
начало
So,
в Мечникове совместно с Институтом вакцин
would like to
Санкт-Петербурга
mention
самостоятельных
the
работ по
beginning
разработке
of
вакцин.
Menshnikov's work
Это лаборатория
together
R&D
with the St.
и новые
Petersburg
цели
Institute of
вакцины
Vaccines in
против
Syria,
трансмиссионных и
where research and
геморрагических
development
лихорадок,
R&D work
денге,
was done
чикунгунья.
with new objectives against
Это и
chikungunya,
прямое привлечение
dengue, and other
университетов.
hemorrhagical and
Нам
infectious
важна
fevers.
хорошая
Also working
лабораторная
directly
и
with the
доклиническая
universities
инфраструктура
involved in the universities.
с
And
собственным
so
виварием.
we really
Мы
need
будем договариваться о
our
правилах
own
этой подготовки
infrastructure
и работы
for clinical
с
studies,
нашими
preclinical
университетами.
work, and so on.
А
And we
это,
will coordinate the
соответственно, и
rules of this
лаборатории,
work with our
и
universities.
специалисты, и новые
And,
требования,
this is
и новая аккредитация во Всемирной
refers
организации
to
здравоохранения.
laboratories, experts,
Конечно,
with
это развитие
new
программы
prerequisites,
бионика, школы кадров
also
и
based
знаний.
on international
Конечно, это
standards. We're
новые
also working
платформы,
on
в
the
частности,
School of Human Resources and
платформа
Knowledge,
производства вакцин
which is a new
на Е-коле.
platform for the production of vaccines
И, безусловно,
based
это ворота
on
российской
Krishna
фармии
coli,
в регион Центральной
and
и Латинской
also
Америки.
the
Как уже говорилось,
Russian
задача создания
pharmacological
хаба
industry
по
in Latin
фарме
America.
и из
And
России,
as
и из
we had
других
said,
стран.
we have the task of creating a hub for other pharmacological products
В частности,
in Russia,
сегодня
here in Central America and Mexico.
мы будем представлять
In particular,
пакет интерфероновых продуктов компании Фирнем
will
и
show
проект
a
на
package
территории
of
лаборатории
interferon
Рамос.
products
Будем обсуждать
and
на полях
a
Конгресса.
project from the
Я
Ramos
и мы с
laboratory
вами
that we're going to
ожидаем
talk about
интереснейшую
here
лекцию нашего коллеги и друга,
at the Congress.
доктора Андрея
And I hope
Симбирцева,
to
с
hear
которым мы очень много работали над
very
развитием
interesting
этих
also
продуктов.
presentation
Это продукты компании Sotex,
Dr. Sirbirsev
эритропоэтин и
on
инексапарин.
the products of
Это
the
продукты
Sotex
компании Bio. Конечно,
product
вакцина от
company
оспы и целый пакет новейших
vaccines
разработок
against
вакцины Пента,
smallpox
Гекса,
and a set of
ботулотоксины.
new beta
Это международные проекты.
vaccines.
Вакцина из
And we will
Китая
also
от вируса
hear
папилломы
about
человека.
international
Это вакцина
products,
из Индии,
the
инактивированная
Chinese vaccine
полиовакцина.
against human
И
papilloma,
также часть препаратов из Индии,
vaccines
в частности
against
инсулины.
poliomyelitis,
Это те
reactivated
задачи, которые
poliomyelitis,
стоят у
and in
лабораторий
particular
и у производства
insulins,
Института
and all
Мечников.
of these
Ну и,
are
безусловно,
part
Мечников,
of the different
как плановая
Menshikov
необходимость,
labs,
должен развиваться
and
и должен
also
расширяться.
Menshikov
Должны
also
быть построены
needs
новые
to
цеха и новые
expand
лаборатории.
its
Но это наш
production
давний
by
план с
building
доктором
new
Роберто Лопесом,
laboratories
и мы
and so
его
on. And that is an
мы его
expansion
в любом
plan that
случае
we've been
внедрим.
talking about with Dr. Roberto Lopez.
И, безусловно,
And
это поиск
we will
институциональных и
certainly
коммерческих
make this plan
инвесторов
a
в развитие
reality.
данных программ.
And without a doubt,
Поверьте,
we also need to
сейчас
work
это
with
средство только в
private
технологии.
institutes and
Ключевые команды
investors
уже
for
есть.
commercial products. Now we will
Ну, и
invest
немного
in technologies.
в
We
своем
have the
институте,
necessary equipment.
который я
And just
возглавляю.
very briefly, my
Безусловно, это моя
institute,
и наша
the
гордость
institute
за
that I direct,
работу с
the
Мечником,
St. Petersburg
институтом,
Institute of
созданным
Vaccines in
правительствами
Syria.
наших
We
стран
are
в Никарагуа,
very
который
proud
сформировал
of it.
платформу
It was created
для
by the
всех
governments.
реальных возможностей внедрения всех
Menshikov
программ,
is
о
now
чем
a
буду говорить и
platform
я, и
for
главное,
the
наши
work done
партнеры
with,
и гости,
our institute
плотно
and the
работающие
government
с
of Nicaragua.
Мечников и НИИФС,
And I am
профессионалы
living
и
proof
специалисты,
of
поверившие в
the
будущее
close
настоящей фармы в стране
cooperation
Никарагуа.
between the St. Petersburg
Это
Institute and
наши
Menshikov, and there are
новые
experts that really
лаборатории
believe in the
по
future of
разработке и
this work
внедрению в
in
создаваемого производства
Nicaragua. So
вакцин,
new
как я уже говорил, на
laboratories
платформе
for
Е-коли.
the
Будет
development
чудесная
of
лекция
vaccines
доктора Севастьяна
based
Рабдано,
on E.
возглавляющего
coli,
эти разработки.
we will be
Это
hearing
полисахаридные
from
вакцины,
Dr.
гемофильная,
Sebastian Rabdano,
пневмококковая,
who is also
менингококковая.
who
Это
worked on different
инвестиции
types
более
of
100 миллионов долларов
pneumococcal and other
из
types
Российского
of
фонда
vaccines.
прямых
This is
инвестиций, что
investment more
очень
than
важно и
$100
значимо для нас.
million
У
from the Russian
Института вакцин
Investment Fund
в России
that are of tremendous importance
сейчас
for us.
стоит просто гигантская программа по развитию, и, безусловно, эти
Institute
вещи
of Vaccine
тождественно
in Syria
связаны
in
с
Russia
Институтом Мечников. Они
has
все
really
будут
been
копироваться,
advancing, and
отражаться
this
и
is
исполняться
clearly
на
related
территории
to
Института
Menshikov, and everything that we
специалистами
do
Института
will be
Мечников. И,
implemented,
безусловно, это
copied and
гриф,
implemented
наш
at the
стержневой продукт,
Menshikov Institute by
флюент
its
тетра
experts,
и
and then,
стандартные
of course,
образцы и сыворотки для Всемирной организации
key products,
здравоохранения.
and
На
so
сегодняшний
on,
день
up to the World
мой
Health
институт
Organization
претендует на
standards.
4-ю
Today,
лабораторию в
my
мире,
institute
которая аттестована российским
will
регулятором,
wish to be
уже
the
аттестована
fourth in
российским
the world,
регулятором, и мы
accredited
подали
by
заявку на
we
аттестацию
are already
в
accredited by
международный
the
регулятор
Russian regulator
Всемирную
and have
организацию
applied
здравоохранения.
to
Ну и,
the
наконец,
World
это
Health
наша новая
Organization
вакцина против ковид.
regulatory
Это новый
commission,
вызов
and
тем
our new
платформам и
vaccine
мировоззрению, которые
against COVID is a
царили
new
до
challenge
сегодня.
that we
Это
have
вакцина
ahead of us.
Конвасел.
And
Я
the
презентовал в прошлом
performance,
году введение
is
ее
a
разработки
new
в этом зале.
vaccine, as
Сегодня
happened
мы
last year, and
уже начали в
presently
России
here
фазу
the information about its
2B/3
development. And
и
today
продолжим
in
в
Russia
Никарагуа
we
в
are in
2023
phase
году.
2B
Ждем интересную лекцию доктора
/3, and
Сергея
we'll be continue
Аракелова,
work
и не
in
только по
2023.
этой
We'll be
вакцине.
hearing from
Более
Sergei
интересно,
Arakilov
по новым подходам в
about
оценке
this,
и идеях
and
профилактики множества заболеваний. Послушаем и
new
поспорим с ним.
approaches for
Это
the
наше партнерство с
prevention
Институтом
and
иммунологии.
treatment. And we will hear when
Целой
we talk with
галереей
him and also with the Institute of
исследований и
Immunology
разработок.
as concerns a gallery really of
Будет
studies.
лекция и в зале, и дискуссия на
And
полях Конгресса
there
с моим другом и
will
партнером,
also,
доктором Мусой Хаитовым.
discussions with
Конечно,
my
это наша
friend
программа
and
«Бионика»,
partner,
хорошо
Dr. Musa
зарекомендовавшее
Haiko,
себя объединение, позволяющее
which
не
is
только подбирать и
who
учить,
heads an
но
important
главное
program
формировать
that allows us
качественно
to
новое мировоззрение
select
у десятков молодых людей.
educate and
Нет более
create a new
профессионального
world vision
и горячо
regarding
любящего
with
свое
young
дело,
people
чем
regarding
доктор
medical
Елена
and
Казакова,
pharmacological
которая
practices.
проведет отдельную
Elena
сессию по
Kazakova
образованию.
is the
Ну и,
person who
наконец,
loves
это
what
выполнение задачи
she's
по
doing, and she
преквалификации
will talk to us
Всемирной
about
организации
education.
здравоохранения
And
наших
we will be
гриппозных
talking about the
вакцин. Напомню, что в
pre-qualification
преквалификации участвует
of our
и Институт вакцины сывороток, и
anti-grip
Институт
vaccines.
Мечников. Мы несмотря на
and also
немного усложнившуюся
our own
ситуацию,
institute.
достаточно последовательны в плотной работе со
Despite the
Всемирной
little bit
организацией
complex
здравоохранения.
situation that we have
В
today
этом
in
году
Russia,
визит экспертов не состоится, но в 2023
and
году мы четко
there
находимся в плане этой
in
работы
the year
и Россия, и
2023,
Никарагуа.
there will be a visit of experts. And
Ну, у нас сегодня и завтра Конгресс будет проходить немного в новом формате. Это должно быть очень
And we will
познавательно,
see a
очень
new,
интересно. Надеюсь, всем очень хорошо
format of the
понравится.
Congress, and
Еще
you'll
раз
get lots of
говорю
new
спасибо
information. I hope you
присутствующим
like it.
коллегам и братьям из Никарагуа.
And again, I
Еще
thank
раз
all
спасибо
of you,
послу
our
Российской
brothers and
Федерации
sisters in
Александру
Nicaragua,
Холикову. И, конечно, добро
the
пожаловать
ambassador
всем
of the Russian
гостям,
Federation,
всем коллегам, всем
Alexander
участникам.
Kolikov,
Начинаем
all
Конгресс.
participants from all countries, and we thank you for your welcome. And with that, I declare this Congress open. Thank you very much.
Agradecemos a todos los miembros de la mesa de presidium por sus palabras. Les invitamos a un receso. Posteriormente con ustedes la primera sesión del panel número 1, nuevos enfoques y plataformas tecnológicas actuales para el desarrollo de vacunas y productos farmacéuticos, estudios preclínicos, clínicos y proceso de registro en condiciones de pandemia. Les invitamos a pasar al Salón Lirio. Tenemos 15 minutos de receso. Muchas gracias.
Salon or room for to a break, a cup of coffee and so on, and we will be back in 15 minutes. Thank you very much.
Uno, dos, tres, uno, dos, tres. Uno, dos, 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 uno, dos. Uno, dos, uno, dos, un, dos, tres. This is the English channel, testing 1, 2, 3. This is just a sound test for the English channel. We are now during the first coffee break. This coffee break will go for 15 minutes. After 15 minutes, we will resume the Congress with the first panel of discussion. In this panel, we will have a keynote presentation titled New Approaches and Current Technology Platforms for Vaccine and Pharmaceutical Development, Preclinical and Clinical Studies and MA Process in the Pandemics. This is the sound test, 1, 2, 3. Again, this is a sound test, 1, 2, 3. We are in a 15-minute break. Thank you.
Probando, probando, probando. 1, 2, 3, 4.
para darle una manito aquí a Iván y seguimos probando para ver si podemos eliminar todo el ruido, literalmente hablando, que está metiendo la cabina de inglés. Seguimos probando a ver si todavía hay esa interferencia y luego iremos a la segunda parte.
Okay.
Ahora alguien está hablando de presidio y se oye nítidamente, está muy bueno. Eso sí, con los audífonos hechizos de don José Daniel Leiva. Siempre probando para ver
Sí.
cuál es la dificultad que
Sí.
aparentemente hay con una interferencia que está siendo causada aquí por la cabina de inglés. Iván está en eso y ya creo que pronto vamos a poder resolver este problema. Ya pasó la primera sesión aquí del séptimo Congreso Rusia-Nicaragua, muy interesante, sobre biotecnología, vacunas, y bueno, como siempre en esos eventos, uno aprende. Y hoy la lección es sobre vacunas, sobre biotecnología, y vamos a aprender sobre el Instituto San Petersburgo, su cooperación con el Instituto Menshnikov aquí en Managua.
Hola, sí.
El Menshnikov es el Instituto Latinoamericano de Biotecnología y el Instituto en San Petersburgo tiene como nombre completo, ya le digo, el St. Petersburg Institute of Vaccines and Sera, o sea, el Instituto de San Petersburgo de Vacunas y Sueros. Y Iván sigue siempre probando. Entonces seguimos intentando resolver el problema que hay aquí. Parece que la cabina de inglés está causando un tipo de interferencia.
Probando ahora este micrófono, a ver si se escucha el micrófono. Se escucha este micrófono. Inglés, se escucha, no se escucha. 1, 2, 3. 1, 2, 3. Se escucha, no se escucha. Sí, no, sí. Ok, ahora vamos a probar el otro micrófono. Un momentito.
Entonces, probando el otro micrófono. From now on, I will speak only in the language of Shakespeare and Gerard Manley Hopkins. As we continue to test, my friends have all suddenly become firemen, and they...
Now testing English 1, 2, 3. Can you hear? ¿Puede escuchar? ¿No escucha? ¿Escucha? Sí, perfecto. Escucha inglés. Borroso. Ok. Borroso. 1, 2, 3, 1, sí, solo está este micrófono ahorita. Ahí se escucha, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, un poco borroso.
Testing. We are testing once again the equipment with now using the other microphone. This one is better. That's because it's hechizo. And although Mr. Leiva looks down upon this humble headset, which I am quickly getting used to actually, and perhaps what's that? For 10 years. So there must be something subconscious. Historic headphones. Which Luis Martinez are we talking about? Yeah, okay, but he's not an interpreter. Do you mean Doña Helen?
No, I mean his company. He will provide
Ah, oh, you got these from him.
this is actually math.
Ah, una herencia. Okay, now I got it. Yeah. Okay. So I have worked many a year and at many, and I'm feeling familiar from the tiempones when there used to be events twice a week, and it's sinking in. Yeah. Right. Did you manage to steal a cup of coffee somewhere? Well, we have to. Well, it is a bit of a. Yeah, it is. Right. The guy's really good, you know. The guy's. Yeah. No. Yep. Yeah, but I mean, to the point where I'm going to have to ask him politely if he works specifically in this field, because you know that in the real world, people work in legal or in other words, we're generalists. Because I mean, when it came to the diseases, and he didn't know what the guy was going to say, he could rattle them off like 4 of them de una sola. Yo estaba captando 2, pues, porque
I think it's probably the case. You know why? Because the market is huge in Russia.
Yeah.
So people will.
In other words, he sounded
will specialize.
like he knew what he was talking about, you know.
So interpreters will specialize.
Yeah.
Those huge markets, they will.
Yeah.
In our little markets where we have
No.
only a handful of interpreters,
Yeah.
no, we have no.
We're generalists, yeah. Which I don't regret, you know. Yeah, as a lifelong thing, I don't regret it. Because if we did only this, our lives would be very boring. You know?
Keeps us busy.
One profession that
Yep.
I have never, you know, for some reason someone told me about it, and
Mhm.
I'm glad I'm not doing that.
Uh-huh.
To be working at the World Coffee Institute in London. So you take the tube from,
Mhm.
you know, your suburb to downtown London. You go up an elevator to the 12th
Yep.
floor, and every fucking day you talk about coffee, you know?
Yep.
That would be really boring.
Unless
days
you
a week,
really like coffee.
8 to 5, unless you really like coffee, you know? So to do that every day, or jam, you know, so it's all about you and Polish jam.
Yes.
Well, after you know the 12 chemicals used in Polish jam, it's
Yes.
pretty, you know, whereas
Yes.
here, it is true, you know, I've always been aware of this. Every event is different.
Yes.
We've seen a little bit of everything.
That's
We
right.
had a veterinarian,
Se escucha mejor, caballero.
pharmacological event.
Ale?
You know, any type of event. It can be agriculture, it can be health. Sometimes it's some.
Ahorita viene en camino el muchacho que me va a traer otro dos. Y estamos pensando que tal vez podría ser qué cosa, el profesor de aquí quiere la última aquí para la transmisora, correcto. Sí, y después yo hice para la transmisora, puede ser. Entonces con Iván, no sé con Iván veamos si podemos poner otro transmisor. Si eso pasa, si no,
So, speaking again so that Don Ivan can figure out what the hell the problem is that we're having. Apparently, we have a situation here where something in this booth is causing interference, and of course these people are a little bit uptight about that, so we're going to have to fix it somehow, and we're working on it. So we continue speaking in English to try to figure out the problem, and continue to test. Let me know if is anybody listening or hearing, to see if there is any interference on the English channel, or am I here just speaking to myself, or are we getting anything done or advancing with the solution to this difficulty, which is strictly technical in nature. So continue. So they seem to think that things have now much improved. They seem to make optimistic like signs. So I think it's been at least temporarily solved.
1. Por favor, pedimos a los compañeros y compañeras que se encuentran en los asientos de la parte de atrás que por favor llenemos los que están aquí adelante. Pueden pasar a ocupar estos lugares que están en la parte de adelante, por favor, para que no queden muy atrás. Vamos a dar inicio, como ya anunciábamos, el panel número 1, que está a cargo de Ángel Balmaceda, que es el jefe del Departamento de Virología del Ministerio de Salud. Marta Rosales, directora de AMS. Carla Delgado, directora de Farmacia.
We will now proceed to the next round, and we will have with us Marta Rosales, Director of NRA Nicaragua, Carla Delgado, Director of Pharmacy, Ministry of Health Nicaragua, Yasmina Umaña, EPI Director, Ministry of Health Nicaragua, Elena Nacharova, Medical Director of the Mechnikov Institute, Vadim Merkulov, Doctor in Medical Sciences, Professor and Deputy Director of the Scientific Research Center for Products for Medical Use from the Ministry of Health in Russia. Vladislav Chestakov, Director of the State Institute of Medicines and Good Practices of the Russian Ministry of Industry and Trade. Musa Kaltov, member of the Russian Academy of Sciences, who is a doctor in medical sciences and director of the Institute of Immunology of the FMBA in Russia. Sebastian Rabdano.
Perfecto. Queda con ustedes entonces la presentación del panel número 1.
So we will now have the presentation of panel number 1.
Уважаемые коллеги, мы начинаем нашу панель, и я хочу представить свой доклад на тему «Переход от борьбы с вирусами с помощью нейтрализующих антител к борьбе с болезнью за счет универсального действия вакцин». Пожалуйста, первый слайд. Так, у нас
The first slide, please. Transition. So, doctor in biological sciences, Mr. Sergei Arakelov. So please show me how I can move through here with the slides. Dear colleagues, please help me with the slides. So Mr. Arakelov will be speaking on the universal scope of vaccines as an alternative to neutralizing antibodies to combat viral diseases. Okay, so a bit of a technical issue here, but let us get underway. So the classic paradigm of vaccines is well known and comes from Genio, who in the early 20th century vaccinated a little boy for smallpox in the 18th century, and with that began a new phase or epoch in the history of medicine. So, and vaccines are, of course, the foundation, the protection of the healthy organism from contagion. Today, we see it as security, a lower risk of contagion and adverse events. So, after vaccinating this young man, he tried to infect him 4 times after 1 year, 5 years, and even more further on, to see if the vaccine was safe and efficient. Efficiency of vaccines in the classic interpretation is used as a model called efficacy, transmission, disease, death. Those are the fundamental indicators. Then the continuation of the action, what happens afterwards, is years and life. No? Will the person live for a short while longer or a long while longer? Will they last maybe 1 year more? Then we would say, well, it didn't work. But today we know that one needs to have a booster shot. And in the classical concept, it takes years or even lifelong. The mechanism includes then adaptive immunity, the B and T cells, the formation of Th1, Th2, CD4, of the balanced response without ADE, neutralizing antibodies. And let us begin with the first one here, and the inclusion of the immunological memory, the B and T cells. Let's start with the first one. Before anything else, I would like to introduce here and say, in the concept of classical vaccines, what we see is that the pandemic had a few peculiarities of the pandemic that makes us change to other concepts. First of all, the highest, which is viral mutagenesis. The second is the absence of viremia, because what is in the blood is neutralized by the antibodies, the virus. But in SARS, no. And SARS goes to the blood, and we can only identify it in the high and low respiratory organs. Then there is a drop in antibody titers, and after suffering from hepatitis B, the person, for example, won't get sick with hepatitis ever again, A and B. But there's also a drop of antibody in SARS, can happen, but may lead or cause a reinfection. There are people who become 3, 4 times, with different strains even. Then there are the systemic lesions outside the actual location of the infection. With SARS, for example, it could be outside of where the infection is, for example, outside of the actual respiratory organs. And in particular, then, there is the post-COVID syndrome, which is also a particularity of SARS-CoV-2. If we talk about the adaptive immune system, here we have the different B cells, plasma cells, antibodies, and so on, and of course the T helpers and T killers cells, with which we move on to the next slide, please. Which is the role of the humoral immunity after the vaccination against COVID-19, which continues to be contradictory today in 2022. If we talk about adaptive immunity, we see that we have neutralizing antibodies who work for a long time against tetanus, rubeola, and so on. But what we're talking about at the beginning, when we still had one strain of Wuhan, we saw that this concept wasn't quite right, and the situation had changed, and so we cannot any longer base ourselves here, in this case, in the classical concept of vaccine. There is also a negative correlation between events and antibody titers between vaccinated health workers with the COVID-19 and the ARNM BNTI62b2. So that's one of the natures, no? According to the Nature, which is a well-known journal. The reactogenicity of the BNT162b2 vaccine against COVID also has adverse effects, and the protection of the antibodies against earlier strains goes down when there are new strains and can reach as low as 0. And in that sense, there is not much neutralizing action. Getting back to vaccination mechanism, we can go to the second phase, which is the formation of Th1, Th2, which is a balanced response without ADE. Next slide, please. I'm pressing the right buttons, but it doesn't seem to want to show. These are different, okay. Two different screens, apparently. Okay. So we're talking about the formation of Th1, Th2 of this mechanism. The Th1 and Th2 markers are different. And based on accumulated experience, we know that the balance of Th1 is more efficacious and less damaging. Adverse effects like allergies or autoimmune diseases, Barré syndrome, and so on, are more related to Th2. And vaccination with integral vaccines can cause a shift to Th2. What other effect is related here to this effect is the expansion of diseases that depend on antibodies. That the virus is connected with antibodies. There are receivers, CD32-1, immune cells, CD32+, monocytes, macrophages, B cells, dendritic cells, and so on. And these will affect the cell. Not always, but there are cases, and we can see from our experience here, in the case of dengue. That's why to date we still don't have a vaccine against dengue. As regards risks of ADE with COVID, there is work that shows us in animals that vaccination always produces displacement to Th2, and so the hamsters in this case fall ill. But the meta-analysis also shows that the population of animals in these vaccines, in terms of population, does not necessarily trigger ADE. Then we are looking at neutralizing micro-neutralization, the reaction to micro-neutralization. Here, when we are in the stage of development, these are done in vitro through the serum of immunized animals. This is important to point out then the following and highlight the following.
Во-первых, в сыворотке нет иммунных клеток. То есть какие недостатки реакции нейтрализации? Мы полагаем, что
so these are the disadvantages of the method. Although we think it's absolutely efficient, but there are no immunitary cells in the serum. So we're not talking here about natural killers or anything. Not all viruses go to the blood. So if we talk about the efficacy of the detection of antibodies, we do it through in vitro. But not all viruses, as I say, go into the blood. And then, if you in vitro, you can, however, differentiate the natural from the non-neutralizing antibodies. In vitro can be protectors of the body, of the organism. So there's a lot of work, not just on COVID, but other infections that demonstrate that the presence of antibodies against certain proteins can be very, very protective against the virus. As regards the neutralizing activity of antibodies in time and regarding the different strains, the results for 2022 show us that the level of neutralizing antibodies disappears very quickly, both in infected and vaccinated persons, persons who already have these in very high titers, antibody titers. For example, Delta, it goes down a lot. It goes down by 2.7 points, and in the case of Omicron, 15.4. No, in Delta 2.7, Omicron 15.4, and that basically reduces efficacy practically to zero. And last but not least, the inclusion of the immunological memory. Here we're talking about memory cells B. B memory cells are defined as cells that are long persistence. They persist in time, sometimes for decades. But for the memory, in terms of immunological memory, these cells are very important. And this is the half of these disappear. No, the previous studies may have significantly overestimated the lifespan of the memory T cells, and the second half of their lives lasts on average only about 6 weeks. So it won't last for a long time because they disappear. The antibodies remain, but the antibodies cannot always protect against transmission from the disease. Regarding immunizing activity, it is something that the bodies can protect us from. If we talk about now, we are the disease itself, the antibody titers and neutralizing activity goes down, but what happens to defenses? The efficacy is also reduced, about 50%, 60 for AstraZeneca, Pfizer or Moderna 65 to 75 after 2 doses, waning to around 10% by 25 weeks after the second dose, and that is also when we began to see new strains, and so the disease recovers, so to speak. And given all that, with the new wave, then we are talking about, the new idea is to create a new concept regarding vaccines. We've been talking about it since 2020, and the World Health Organization is talking about it now, but the concept in 2020, and it is based on the following. First, the idea is to pass from superficial proteins to internal proteins, because there are almost no mutations. Secondly, to move from neutralizing from antibodies for the virus to cells against cells. So where the immunity itself protects the cells. Third, to move from adaptive immunity to innate immunity. And the fourth would be to move from a vaccine against virus toward a vaccine against the disease. Not all my colleagues are in agreement on this interpretation, and I'm going to get back to this a little bit further on. It doesn't mean we're trying to fight the disease in the infected person. We're still talking about about prevention. But it can be the prevention of the disease or the prevention of the development of a more serious disease. So we're looking at transmission and the really damaging effects that the coronavirus virus may cause. Now, why did we decide on nucleoproteins or protein N? It is because it's known for its immunogenous characteristics. Almost all negative RNA proteins have this quality. And Dr. Lvov, for example, worked on this research on cell immunity. It's a conservative protein that is not very flexible in the strain. There are publications from the year 2020 about using it as a candidate for the new vaccine. And the reason is because the S protein and the model for the protein S vaccines instead of what we call the whack-a-mole model, you know, we need to really hit it hard, no? And it focuses vaccination efforts on a single spike variant. And some say that that the N protein is a good candidate for the development of a new vaccine. In January 2022, the OMS called on or pointed to research and encouraged research that looks at the different mechanisms against all the different strains, you know, the dual spike and the nucleocapsid mRNA vaccination, because what we're seeing is that the different strains are coming out for different areas. There's now 4 or 5 very different strains, and it's highly unpredictable. So I think we can do something, you know, the same model sort of with influenza, when, you know, the seasonal strains. But with COVID, unfortunately, that doesn't work. It's impossible. So instead of whacking the mole in the game here with the little boy, the next phase of the concept of Convacel is to move from instead of vaccines against viruses, cells versus cells. The immunological theory for Convacel is based on the immunological theory of vaccine action based on protein N. And this is not an absolute model. You know, it's conditioned with to the antibodies, and it's a well-known mechanisms, natural killers through antibodies. This combines the different adaptive immunities, and the role of innate immune deficiencies are associated with severity and poor prognosis in patients with COVID-19. Anthrax, for example, or certain pests or plagues that attack animals. But here we're talking about innate immunity. It works without being covered by a vaccine. So the adaptive immunity is that you need to face up and deal with the disease to put its defense into action. Another peculiarity of innate immune deficiencies shows that those people who work with animals have immunity against anthrax, and this innate immunity protects them not so much against the infection, but against the disease as such. And that is why innate immunity somehow brings us, you know, back to what we are talking about here. Namely, you know, for example, the T lymphocytes or macrophages, neutrophils, basophiles, eosinophiles, all of these are a combination that use innate immunity to protect us. And the fourth part, again, from vaccine to virus to vaccine against the disease, and this is something that is well known. COVID-19, the research on COVID-19 show us vaccines are effective at preventing severe disease, hospitalization, and death, but not the transmission as such of the disease. Then we found out that the hospitalizations and the severe disease was not because of the vaccine that people were being protected. As the vaccines that we have, nobody knew that they had actually already existed. For example, BCG vaccines, you know, it's not that effective about transmission of the disease, but it defends us quite well from more severe forms of TB, such as meningitis. There is also something called Shingrix, which is a recombinant vaccine against herpes zoster. It doesn't prevent the infection, nor does it have neutralizing antibodies. However, Shingrix does reduce the risk of developing shingles in almost 97% of people 50 years or older. So it's quite efficient in its own way, although it doesn't have certain characteristics we thought was important. So we're learning a lot, and here there's new work being done, the role of nucleoprotein in immunity to human negative stranded RNA viruses. And here, different viruses that you can see on the screen, and they all have the N protein, the little green chain you're seeing there, that's the N protein. Earlier analysis shows that this protein facilitates an adaptive immunity against several types of infections, and this is why it could be a new concept and a vaccine in the new generations here. And in the work we're doing, we are trying to develop a vaccine against dengue. One of my colleagues is going to talk about that. It's been going on, studies and so on, to create a vaccine against dengue. But many, many obstacles in the way. For example, in the Philippines, when testing the Zonafi, several people died, including children. And so what we're doing here, we are using a different approach to use these new proteins, such as protein N, to see if they could possibly be effective. And with that, concluding my presentation, I give the floor to a colleague of mine. Just a very, very brief technical recess here, or break, because we need to connect to our colleagues.
Так, микрофон не работает. Хорошо слышно, да? Все нормально? Так. Спасибо. О, прошу прощения. Итак, коллеги, мы начинаем нашу сессию, дискуссионную сессию. И я хочу предоставить первое слово доктору
Good morning to all. I would like to give the floor to Dr. Angel Balmaceda, the Director of the Department of Virology of the Ministry of Health of Nicaragua.
Dr. Balmaceda,
So, Dr. Balmaceda, I would like to ask you to tell us how you are looking at how the pandemic has influenced and had an impact on your regulating agency and on your department. So if you would like to tell us a little bit about that, Mr. Balmaceda. Good morning, and thank you very much for the invitation. I am Mr. Angel Balmaceda, and I am the head of the Nicaraguan regulatory authority, and in the CNDR, the Ministry of Health, the regulatory unit of the Ministry of Health in Nicaragua. And what we here call PCF in real time, the Ministry of Health or PCR has experience in diagnostic and molecular technology. For example, we've been even pioneers, I would say, in this regard. In the 90s, the laboratory was the first that applied this when we had localized a localized dengue pandemic or epidemic here in Nicaragua, and we were pioneers in the diagnostic in influenza, and by 2009 we were already using a PCR technology in real time. I would like to say that with that, when the SARS COVID began, we were actually prepared. And by February of 2020, first case in Nicaragua, which was identified in March of 2020, but already in February, a month before that, we had the reagents for this kind of disease. And then we were trained by the Pan American Health Organization, and the reagents, I mean, the main problem was the sustainability of diagnoses, because we needed to acquire then the reagents, and in the end we got these through donations, purchases using the national budget. This allowed us then to satisfy the demand for diagnostics in this regard. What was, in fact, a challenge for us and for the health authorities, to answer your question, Doctor, was the genomic sequence to identify the different strains as they developed during this period of time. And to that end, in 2021, we had to send samples to the University of Michigan. They're colleagues of ours, and by May or June of last year, we were trained online in genetic sequences, and based on that, we started to do our work to be able to do a genomic of all strains, and we now have the technology well enough developed so we can have genes monitoring and genome sequencing, and both in space and time. And this genome sequence is going on now throughout the country, and there is real-time surveillance. We do the sequencing, not just for SARS-2, but also other viruses, such as dengue. We know the phenotypes that are circulating and so on. So our future is to keep this sustainable, of this genome sequencing, and do this throughout the country, both in the health services, epidemiology, and so on, so that they can alert us if there are new strains that may cause a public health emergency. We have also been doing studies among the population of the vaccines that have been applied in the country. We know, and in developing country, it was very, very difficult here to get vaccines. So the health authorities had to vaccinate people, but only according to availability. And we depended on donations, so people had been vaccinated with sort of a cocktail of vaccines, the different types of vaccines in different parts of the country. So we used serological methods to determine the antibodies of the population, and found, and above all and specifically in districts 6 and 7 of Managua, and found that the population in the country has high levels of antibodies, as you know, and it's very interesting. Your presentation was very interesting. These are not determined by some particular technique. We cannot say specifically if they're neutralizing or not, but the system is aimed against protein S, and there's some variation between the anti-spike proteins and the neutralizing antibodies. So what we can extrapolate that at least at the point in time that we took the sample, which was January, February this year, the population had high levels of antibodies. So we think that in this regard, and we're at the laboratory, we think that, and which is the part that I direct, the unit that I guide at the Ministry of Health, we're prepared for future challenges. And of course, hopefully not some very terrible virus, but the situation as we have it now is basically under control. Thank you very much.
Поскольку наш регламент достаточно уже сжат, предлагаю перейти на обсуждение 10-минутное.
I then propose that we not to speak maybe more than each person 10 minutes. But what was said about the diagnostic, that refers to diseases. We know that in the pandemic, a classic approach to the real disease was changed, and we now know that if there is resistance, we know there's resistance whether it works well or not. So we went to monoclonal bodies, and it seemed that this worked well at the beginning. But it then turned out that the monoclonal were not working well because the strains were changing. So then we asked ourselves, but are there any platforms that can be universal? Well, now I give the floor to Dr. Haitaf, the director of the Institute of Immunology of the Academy of Sciences, and he will tell us about one of these platforms that exist and is already being applied. Thank you very much, Sergey Alexandrovich. I would like to begin by greeting my dear colleague.
It is for me to be a participant of this 7th Congress, Russia in Nicaragua. This is the second time that I am visiting the wonderful and welcoming land of Nicaragua. So thank you very much to the organizers of this event for inviting me, and also to the company that also invited us here. Yes, Sergey Alexander, you brought up a very important topic today. For the past 2 years ago, we have been under this COVID-19 pandemic, and despite the successes that we have had, we also realized that the virus hasn't stopped mutating, and the efficacy of the vaccines is decreasing, and the medicine is the effectiveness of the medicine is also decreasing. So we need to produce efficient, safe medicine that is not depending upon the mutations of the virus. This is a unique development that we created in our institute, along with Pevenitz, the Meshnikov Institute. This medicine was created by an order by the Federal Medical Agency, Solar, and it was supported by the government of the Russian Federation. In the past, I told you about the Mir-19 medicine. I will not tell you too much about this specific testing, or I will tell you very briefly about the preclinical trials. It is an antiviral medicine that is unique, and it's working based on the interference mechanism for RNA. It protects the materials. With this mechanism, we can stop the expression of any target gene. Why smear? It's low interference, or MIR in Russian, MIR. It will be more efficient with the low interfering RNA, which is affecting the virus's genome for SARS-CoV-2. When we are inhaling it, if it's inhaled, it's going through the path that the virus expands or enters the body. So by doing this, we block its replication, we block the infection, and we saw all this in the preclinical trials, and we have proven that Mir-19 reduces over 10,000 times the viral charge, as we saw in the #1 issue of the preclinical trials. Then we moved on to the clinical trials of this medicine, and during the first phase we proved that it is absolutely harmless for human life. It's not a threat for human life, and this allowed us to move on to the second phase, where we saw patients who had the disease, and we were comparing that with the medicine that was at the time being recommended for that specific purpose. The group being treated, we had a group being treated by Mir-19, and we had another one that was using the medicine recommended by our Ministry of Health. We proved with a lot of substantial evidence that it is highly efficient. Mir-19 is safe. It reduces the several affections and, of course, the death rate. We have also proven that complete healing in severe to moderate patients took place days earlier than the mainstream group. So we are very thankful to the regulators who were able to understand us and who really understood that it was important to have their support in the development of this medicine. We're very grateful to them because of that. Today, this medicine is registered in the Russian Federation. It targets COVID-19 treatments for medium to severe cases of COVID-19. And we then moved on to this phase 2 and phase 2B, which we have completed. We submitted to the regulators the new aspects that were to be registered, and I'm presuming that in the next session I can tell you more about our successes. I would like to highlight that in a very short amount of time, only 2 years, with the support of our CPBNIF friends and also the Microbiological Agency of Russia, we went from the idea of the medicine to proving its efficacy and safety that is now circulating or being distributed among the population in only 2 years. And now we are only waiting for the indications or the instructions for the administration of this medicine. This medicine was created in a platform, in the peptide platform. We also have treatments for asthma and rhinitis. We have completed the clinical trials and pre-clinical trials, and next year we will move on to the clinical trials. And next year I'm sure that I will tell you more about our successes, not only with MIR-19 but also with other medicine that is being developed through RNA interference. Thank you very much. And I would just like to add that we are working very closely with the Immunology Institute, not only for the preclinical and clinical trials, but also with the St. Petersburg Institute, that is also producing this medicine today. As for the universal platforms, the RNA platforms are new, but there's another well-known platform that is even more widely spread, which is interferon. Interferon is efficient. It's not efficient in all the cases, but this platform adds validity, and in light of new challenges, we need to pay attention to this medicine, which is one of the most known medicines. Now I'll give the floor to Andrei Sirvitsa, who is the Chief of Scientific Research, who can tell us more about the developments of interferon. Thank you very much, dear Sergey. And it's not just about a platform, but it's a group of medicines that we are introducing actively. At a global scale, we see that the chemical pharmaceuticals are only efficient to one virus in most cases. Antibacteria medicine is introducing new antibiotics. We see very limited number of medicines in neutralizing antibodies are also another aspect. So we see that some medicine that was effective against the first versions of the virus, but now it stopped neutralizing new coronavirus strains that are coming up. There are also certain limitations in interferon as antiviral medicine is very unique because first it is our own production, is endogenous, it's antiviral, is a wide range, and another important aspect of this is that it is effective against DNA and RNA of the viruses. When interferon was discovered 60 years ago, it was quite interesting because specialists were analyzing the contact of the cells with the strain, and they realized that after the contact with the strain of the virus, they were showing resistance to other strains. And precisely that phenomenon was called the interference in the cultivation media. The first virus was showing an antiviral effect, and they called this interferon. And because of this unique effect, it was very clear that interferon could be induced by a virus. And it was uniquely antiviral product, and it's something that nature has given us to face viruses. So what can we do now? Now we can reproduce the molecules by using genetic engineering methods, and we can get a full copy of the antiviral antigen of our cells, and based on its product that we have developed interferon products a few years ago, and there's a number of protocols and a number of viral diseases that can be treated with interferon. Now, with the case of COVID-19, with serious cases of flu and other viruses, we can use the intranasal interferon forms, and it's a very important platform that is adding new approaches, is an antigen, and it's widely used. It can be administered directly in the respiratory airways. It basically sprays or drops, and we have acquired a new technology in our institute that also allows us to create aerosols or sprays so that it can be better administered in the airway. There's also a number of intranasal medicine that we have also created in Nicaragua. In the Meshnikov Institute, we want the institute to have access to this so that it is also accessible to the Nicaraguan market. So hopefully we can do this soon. Thank you very much, dear Andrei, and I am very sure that we will pull it off because we are supporting each other, we're working closely with one another, and we want to pursue universality because it's both based on RNA and DNA, but I would like to also use universal technology platforms. I'd like to give the word to Sebastián Rabdano, who is the chief of the research center of the St. Petersburg Institute. Thank you.
Спасибо, Сергей Александрович. Спасибо организаторам.
Thank you very much, Sergey, and for giving me the floor. Let me just briefly tell you about an approach that is pretty much down the same lines of what we've just been talking about, universal vaccines that can be created based on technological platforms manufactured in large amounts to deal with pandemics in different countries. The COVID-19 pandemic has shown us that is a need that can happen suddenly, and the demand for a particular vaccine can grow multiple times in a very short period. And to satisfy this demand, it is necessary to use a technological platform that allows us to manufacture a number of a particular pharmacological product that responds to all the different security measures and so on in a short period of time. But you have to have the platform ready, and when we create it, you need to think of controlling all the different details, all the components of this platform. Right now, at the moment, and I think everybody would agree on the possible platforms for the fabrication of these vaccines. But I want to mention recombinant proteins, because this platform allows for the control of the makeup of the vaccine, both from a point of view of its chemical composition as well as the makeup of the antigens. We have the possibility of controlling them using genetic engineering, the sequencing, and the epitopic sequence and so on allows us to adjust the vaccine with great flexibility to ensure its efficacy. And thinking of the universality of the use of this vaccine can respond then to this challenge. First, we need to think of, why are vaccines today not universal? I think that it's the evolution of the virus itself is the driving force that is, making it so difficult. It is the main challenge. And the virus tries to find new ways to cause contagion in the carrier, and at the same time, it needs to keep certain parts that are intact and ensure its functional viability. Because the life cycle of any virus, there are phases that it needs to comply with, needs to live through. Otherwise, its life cycle will be interrupted. And if we, by developing a vaccine, concentrate on those parts of the vaccine that don't change in the course of its evolution, then we have the possibility of coming up with a universal medicine, a universal vaccine. And a good example is the Congreso vaccine, which takes advantage of a protein that is highly conservative. On 30 September, we published a study in which we showed that even in the new SARS-CoV-2 strains, we could see, according to the traditional metrics for the measurement of immunitary response, according to many indicators, the response to the different strains, including Omicron, in the case of people vaccinated with Congreso, was the same or even higher than those who had the disease naturally. So it is a universality that has to do with the selection of the conservative epitopes, and we think it is a very efficient approach. And having this platform and this design, we are working on new products. We did several studies, and the first thing I would like to mention is the vaccine against dengue. As we've said earlier, it is a very common disease caused by a dengue virus, and there's different types of this disease. but we see that at a global scale, there are at least 4 serotypes, and we need a universal vaccine, one that is safe and that has all the characteristics mentioned by Sergey at the beginning of his platform. And the platform I'm talking about and the approach I'm talking about are optimal to soon be obtaining results. I think that by next year, we'll have the first results for the research that is being done in the Menshnikov laboratory here in Nicaragua. And by the middle of next year or so, we should have quite clear what composition of a vaccine we're going to recommend to obtain a technological platform to basically have then in place a vaccine against dengue. Thank you, dear Sebastian. I would like to, if I may, just add a little bit to what you said about the platform, which is the use of E. coli as a platform. No, it's been well proved. We know that it has no risk. And if we're talking about vectors and so on, there's a lot still to study regarding safety of the results in the long term. But when we talk about E. coli or interferon, these are proteins that are easily available, can easily be cleaned, and they don't include, complicated genetic information. So it would be a universal platform that will allow us to deal with the new challenges. It's easy to scale up, and based on these, we can achieve significant volumes of these proteins, and we need to have these ready. And with that, I would like to give the floor to Elena, and she will be telling us, Elena Nacharova, who is the medical manager of the Menshnikov Institute, and her department has done excellent work and perfect work, in fact. So, Elena, the question I want to ask you is, what new methods have you used when you had to work urgently on these new vaccines for the pandemic? But I mean, in this very limited time, you had only really a few months to come up with some sort of vaccine. How did you do it?
Меня слышно? Слышно.
Well, I think we can hear her.
Еще раз. Добрый день. Сергей Александрович, спасибо за представление.
Good day to
И спасибо
all.
за ваш вопрос.
Thank you for your
Хотелось бы отметить, что
introduction,
в момент объявления
Sergey,
пандемии в марте
and
2020
I would
года
like
у нас
to
проводились
underline that in
исследования
March of
клинические,
2020, when the
и
pandemic
уже тогда для нас было
began,
вызовом
we
объявленные
did
условия.
clinical studies. We had already been doing, and it was a challenge
Пришлось
for
на
us.
время
Some
остановить,
studies
поскольку
had
было
to
непонятно,
be
как действовать
suspended for a
в
while
сложившейся
because
обстановке локдауна,
we
но постепенно
weren't clear
все
on how
научились,
to
ввелись
proceed,
такие параметры,
but little by
удаленный
little,
мониторинг,
we
все
have found
перешли на
new
использование
tools,
электронной индивидуальной
introducing an
идентификационной карты,
important
стали
long-distance work
использоваться
component
подход в мониторинге
using
клинических
individual
исследований,
electronic
основанный на рисках,
devices,
называемый risk-based мониторинг,
the
и тем
use
самым
of special
исследования и других
platforms
препаратов смогли
to
продолжиться. В том числе, что касается
do
проведения
work at
доклинических
distance,
исследований к особенностям
and
относились
regarding
это необходимость использования
preclinical
новых
studies,
видов
the
животных,
need arose
которые не особо
to
были
use
распространены и
new
популярны
animals, new types of
до
animals that
объявления пандемии. Для начала необходимо было определить модельный
didn't
вид
really exist in our region
животного,
before
и выбор
the pandemic,
был
and
обоснован
we
тем, что
need to
заболевание
select
развивается у
these,
хомячков
the
золотистых,
search
а у других
of
животных,
those
которые классически применяются
that
в
reflected the
оценке
characteristics
специфической
of the
активности
disease.
вакцинных препаратов,
These
патология не
were
развивалась. Кроме того,
specific
для
animals we needed to control
оценки
the effect of
клеточного
the vaccines
ответа
for the new
мы использовали
pathologies
иммунизированных животных.
and
Исследования проводились при
to
помощи
evaluate
коллеги
the
Себастьяна
primary
Олеговича, и они
response.
проводились в
We
лаборатории в США расположенной.
used batches of
Для того, чтобы
animals
в рамках
we
выхода
were
на
bringing in
клинические
from other
исследования мы
countries
уже понимали, какой механизм действия нашего
and
препарата.
also
И,
getting
как сказал
help
Сергей
from
Александрович в
laboratories in the
своем
United States.
докладе,
And
необходимо
Sergey has
было
very,
доказать
very
механизм
helpful,
действия новый, и это было доказано
and
в
all of
рамках
that
проведения
helped
клинических
us
исследований,
to test
что у нас
the
не только
effect
гуморальный
of the
иммунный
clinical
ответ срабатывает
studies
клеточный, и за счет этого происходит
control
элиминация
the
вируса.
humanitarian response,
А что касается
see how the
возможности, как
replication
это было
of
все
the
реализовано,
virus was
сплоченная
developing
команда
in the
коллег,
immunized animals, and we were
стремление к
able to
цели
overcome the challenges
и вера в
due
результат.
to the, we understood
Вот,
that we need to
спасибо.
work quickly and together, but it was really good teamwork. Thank you, Elena. I think the work done by Menshnikov was really good, and we saw animals, non-traditional animals, shall we call them, not just the usual rabbits and rats, and we saw that the vaccine was working, and it was a great surprise. And we realize we need to work more systemically and see the different animals, not always just sort of hamsters and mice. And in this case, we used hamsters, but there's things that doesn't work with some animals, works with others. So we learned a lot there. And in the case of hepatitis and so on, we haven't yet been able to, we had to go to clinical studies without doing any studies on animals because there was no possibility. But anyway, that's just an anecdote. And now I would like to give the floor to Elena Nacharovna, who the medical director of the Mechnikov Institute, and she will be working in Nicaragua. And of course, there's some additional complexities and challenges on that front. So when one working at home, you have things at hand. Here you had to, in many ways, start from scratch. And so maybe you can tell us also a little about that and the results, Elena. What can you tell us about your work and maybe share your vision of the new challenges and how you see the future?
Thank you very much for your question. In 2021, we conducted clinical trials, multi-centric, for the trainings by WHO, for the pre-qualified vaccine from the Republic of Korea, but it was not the first study that we did in the Republic of Nicaragua, because before we did it with the University UNAN in León. We also conducted research, and we did pharmacovigilance along with the authorities of the Republic of Nicaragua, and we also prepared for large-scale clinical studies. We were working on that for almost a year, and we were working closely with the Autonomous University in Managua. We wanted to also create a contract-based organization, and in the end we changed the method. We changed our minds on how to do it due to different aspects. Among those, we had to reassess the scenario. We were facing the fact that we were doing the studies during the pandemic. It was not only about the pandemic itself. There were many patients that wanted to reduce the number of visits to the health units, and also we also had a vaccination campaign that was also happening at that time. So the volunteers who could have been volunteers for our clinical trial, they preferred to get vaccinated against COVID-19. And that was a bit of a problem to find patients as volunteers for this study. But we made it through. What I would like to highlight about the clinical trials in Nicaragua is the following. Today, we can say that in Nicaragua we have a really good infrastructure to do these types of studies. The institutions are very interested in this, mainly the Ministry of Health and the regulatory agency or authority by the Ministry of Health, because they issue the certifications for the clinical trials. Among all the presentations that I've seen today, we can see that there's a lot of innovative medicine that is being created, that is being produced, that it is not in keeping with current standards, like titer antibody, as we did with other vaccines. So we need to apply different approaches, different methods. MiR-19 cannot be assessed under conventional or mainstream standards, which makes it more important for regulatory authorities to find new criteria to detect or identify the efficacy of new medicines. So I can certainly say that the regulatory entity in Nicaragua has the full capacity for the clinical trials to be adaptive. And we are going in the right direction regarding clinical trials. We went through SECMED's supervision, and SECMED is the regulatory authority of Cuba. We also got a really good rating from SECMED and also from an independent committee, also from the ethical committee, and this ethical committee is based in the Autonomous University of Managua, and the ethical independent committee is in charge of assessing clinical trials, and they have the ability to bring specialists from different spheres, immunologists, special statistics people, specialists in immunology, in order to conduct a correct assessment of these clinical trials. When it comes to especially the ethical aspects of immunizations, and all I can do is praise the institutions that carried out this work, because they worked hard and they really put an effort to work with the volunteers. The healthcare system that we have in Nicaragua enables a good and unique contact among researchers and between researchers and volunteers, because volunteers have really trust doctors, and this allows us to create a database for future a database of volunteers for future studies, and also it allows us to strengthen the trust by the volunteers, by the patients in the doctors and the healthcare systems. And the studies go for as long as 6 months, and we need volunteers to be committed to the study for during this time period. The involvement of doctors in Nicaragua is also important. Despite the work with volunteers is almost over, doctors are still working in the framework of this research. We have the support of about 150 researchers, and they are building their professional capacities. And we did the last training probably about 1 or 2 weeks ago, and it was training on best practices, and it was held at the National University in Managua. And we are keeping a good methodological knowledge, and especially among doctors or medical staff in Nicaragua. In the last study, we had the support from 7 institutions, including, and we had one researcher in chief in each one of the units, and we also had the support of patients. We worked with the laboratory of the hospital Vélez Pais, and this really helped us conduct a more centralized study. When it comes to the challenges that we faced, I would say that they were the creation of an electronic database and carrying out or implementing the monitoring. This is, of course, a very important aspect, and it entails risks. And the monitoring organization, for instance, online monitoring was not only carried out in Nicaragua, but also we brought auditors who visited the country in person, and we also had other experts who did it online. Another challenge that we faced was finding a medicine to be compared against, because double blind. So it's very important to have the participation of the volunteers and also the participation of the Meshnikov Institute, the organizers, the Ministry of Health, the National University of Managua, and only through joint collaboration between all the entities in the study will ensure the successful attainment. To conclude, I would like to say that Nicaragua has the potential. Nicaragua has the potential to continue to conduct clinical trials because Nicaragua has now all the necessary infrastructure. It's important to highlight that the people are eager to continue to work. I would like now to give the floor to our brothers from Cuba, Olga Lidia Casanueva. I think that she is connected. Is she online? No, she's not. She's from SECMED Cuba. And now let's move on to the next presentation or next speaker, Marta Rosales, who is the director of ANRF, and Carla Delgado, who is the pharmacy director of the Ministry of Health of Nicaragua. The question is, how were you able to materialize this emergency use authorization or the emergency registrar, because it was a challenge, because you had to be flexible. What's your experience?
Buenos días a todos. Primeramente, pues, gracias por la invitación a participar al parte de la autoridad reguladora. Y si bien es cierto, como decía el colega, para la autoridad reguladora, no solo para Nicaragua, sino para las
not only for Nicaragua, but for Central America, and the experience we've had with them is a very important challenge and has been a lot of hard work. And to be able to issue emergency use licenses for these different vaccines. But we were able to do so, and by doing so, the population was then vaccinated. The national authority, the National Vaccine Authority, because there's regulations, laws, and so on, and there were minimal requirements that we, as regulating authority in Nicaragua, had. We then, and indeed we were then created legally by the National Assembly or Nicaraguan Parliament with the National Authority for Sanitary Regulation, and there the emergency use of other led to us as a national authority to regulate and facilitate on a way to becoming recognized, no, and gain the trust also of the top authorities, so that we can deal with this kind of emergency situation. Another challenge that we had as an authority was to simply develop more active surveillance, an entire system of pharmacological product surveillance. And as a national regulating authority, we then created the National Authority for Sanitary Regulations, so that we could deal with the assays and the studies and the use of the different medicines to treat COVID. Likewise, we developed then a number of clinical essays that allowed us as a regulating authority to establish what the requirements are for the evaluation of these clinical essays, and see the results and evaluate these for the obtention then of the permission for emergency use. As Elena said, we're part of a review committee at CONIS, and which is the commission that works and students on the supervision of research. And a research or challenge was, of course, access to medicines, and of course at the beginning the vaccines themselves, because we needed first to have the regulatory authority allow the medicine to be used, or the vaccines, and then a team that would be able to evaluate and allow the emergency use. And we had to really act quickly to see what or challenges would be, or criteria would be, for the emergency use of the vaccine. Regarding pharmacological surveillance, we also coordinated with something called PIES, which is the expanded immunological survey, and then also specific follow-up, if there's a notification of the appearance of a disease somewhere. in the country. And I would like to give the floor to Dr. Marvin Villalta, who is the person in charge of pharmacological surveillance, to expand a little bit more on how we dealt with the situation. Thank you. Then another thing that we did as a regulatory authority was, we had to be more active, as the doctor who preceded me just said, and to be able to deploy the vaccines at national level. And so this pharmacovigilance is something that we really had to, get underway, and there were, so that we could see adverse events happening and give it continuity. And so this pharmacovigilance took place, and we were able to take the strategic guidelines to develop this surveillance and vigilance of other public health issues. And we, as a regulating authority, belong to the National Committee for Research in Medicine, and we belong to this association, and therefore we had to also be providing technical support to anything that is done clinically in the country. As Ms. Miesznikov said, we have developed on working with all of the researchers who have trusted in our country and have come here to help us, and we are, of course, cooperating with them. So now I give the floor to Mr. Dr. Merkulov, and Deputy Director of the Scientific Research Center for Products for Medical Use of the Ministry of Health of Russia. He too had a major challenge at the beginning because he too had to get an entirely new approach at the beginning, and they created the first vaccine, Sputnik, and then a number of medicines, and this had to be done really quickly. So one has to highlight the great amount of work done by Dr. Merkulov in the development of a number of vaccines to be able to counteract the pandemic. Good morning, dear friends. I am always a pleasure to get back home, but I'm also participating in this Congress for the 6th time. I'm sorry that I was not able to participate in the first, but always very interesting, these Russia and Nicaragua Congresses. And not just on the regulatory issue, but also in terms of biotechnology, molecular technology, and so on. We understand that
она велика с точки зрения обеспечения здравоохранения в условиях
and also public health under conditions when in the case of COVID, one is, let's say, not prepared for something like this. And because really COVID was like an engine, really, almost like a locomotive that got us to, that made us all of a sudden medicines on which we are just dreaming at one point, and thinking maybe we'll get it in a few years or even decades, and suddenly we were able to make them marketable in a year or two. Was just absolutely amazing. And we're very pleased here this morning already, hearing from the first person who spoke here, when our colleague was telling us about the diagnostic system that was deployed in such a short period of time, and not just the diagnostic, but based on the classic serological assays, but also and PCR, but modern state-of-the-art technologies in real time. And what they did in their labs rapidly is really important. Why? It's important because to cure diseases such as COVID, start with an effective diagnostic. If you don't have a good diagnostic, then it's like we're blind. We don't know what to cure. We wouldn't know what to cure. So, and diseases that come through viruses, in particular respiratory infections, is difficult. They're difficult to differentiate. They're all basically using the same mask. And so using really modern diagnostic systems, if we talk about infectious diseases, there are several different phases in prevention. The first is long-term prevention, as in through vaccines. COVID has allowed us to approve in a brief amount of time a number of medicines. We're talking about here about a platform, for example. Platform. Why the platform approach? Why is it so important? Because any assay is really an enormous cost for the state and for public health. And when you have a platform approach that is already formulated, then you can quickly, flexibly, and dynamically change the potential targets. You change the virus at whom you are targeting, and then you can attack with proven efficacy. There are other infections as well from a point of view of public health. What Dr. Arakelov said at the end, in perspective, we are looking at, if we, in the case of Nicaragua, a vaccine against dengue, that would be a very, very, very pertinent issue here in Nicaragua. And with an approach that guarantees efficacy and safety is really important to us as regulators. And of course, we pay a lot of attention to these details. And if we can get a quick vaccine on dengue, and short-term prevention, no quick treatment with monoclonal bodies, then they have demonstrated their efficacy in the first stage of the epidemic, and then they became less effective, and we could see why. Because the virus was genetically changing, and different approaches and platforms, different pharmacological products that are effective at prevention and are good in the short term, but we need one that is not just the short term, but also the medium and long term. And here's monoclonal bodies did very well. And I'm talking about the consequences that emerge when the disease becomes severe and the person becomes very ill. And then monoclonal bodies help in the next stage, and that is then the great enemy. I always say virus is the strongest enemy of humanity. It's the most dangerous because it's invisible. And we're talking here then about therapy, the struggle against the virus, different pharmacological products, and all of those I have mentioned. They were highly effective, but not ideal, because there are no ideal medicines. And we hope, of course, to find a universal but panacea is still a dream. To get universal vaccines would be a panacea, still a dream, but sometimes dreams come true. And these were created at different stages of protection, prevention, and treatment. And I forgot also to tell you, now we are in a different stage. We've all had it at some point, this COVID, and now there's consequences. There is a concept, long COVID, when the disease is over, but the consequences stay with the person. And these are of different genesis, of different reasons for this. And there is a line then of applicable medicines with different degrees of efficacy. And there's many platforms that we're talking about here today. And some of them, of which my colleagues were talking about, were prepared in record time, practically, due to the fact that it really was a biological emergency, and then because our regulatory authorities were open and flexible, we were able to act quickly and can today share with you these experiences, and now these products can be used in your countries. Just to wrap up here, but yes, if all of these extraordinary achievements through the platforms required decision-making from regulating agencies quickly, time was of the essence. We had to always be a step ahead of events. And what was a real lifesaver for us was the emergency use approach. And what does it mean? It doesn't mean you just say yes, yes, yes to everything, but if those that have deficient efficacy cannot be allowed out into the market, so they need to be effective, safe, and also pharmacovigilance, as my colleague was saying, because the conditions under which they use, if you don't have a third phase, which is the pharmacovigilance, then you're taking a great risk. So this is really important. And the first sample or assay that shows that if you see that it's not so effective, you can quickly also take it out again if it's not working. If it's a challenge, it's a challenge for regulatory science. It is a challenge for the community of experts, rather. And it was very interesting in these past 2 years because these were advanced assays, and these were carried out by high-level experts, really first-rate scientists. And decisions had to be taken quickly by the Ministry of Health, by the regulators regarding the use of these different pharmacological products. And now we go to the next product, which is to think about this, think about what happened, think about what was done, see what pharmacological products are effective, what platforms we can use to formulate new products, not only for the treatment and prevention of new diseases, but also the prevention and treatment of new diseases, oncological, genetic, and so on. But the important thing is, and I don't want to talk about other countries in Central America, but, I do have to say that in Nicaragua there is a research center, and that is the Mechnikov. And there we can do different assays, work with the regulators, develop new pharmacological products, and create really good medicines that can be used, and that's something from other countries in the region can also learn. And that is what we want in the end, that we all cooperate in only one place and then create other centers in other countries. Once the experience in Nicaragua. Science doesn't end. Science keeps going, and the regulatory work as well. And the hope is that one day there will be 1, 2, 3 Menshnikovs in different countries as well, in other countries of Central America. 7 years ago, when we just got to and met for the first time, we traced the different routes, and now we've gone down the road. Now we can turn back, look back, and see how the seeds that we sowed 7 years ago have grown. I really am happy and pleased that you invite me to each of these congresses. As I said, I've been to 6 out of 7. I'm always comfortable in Nicaragua. I'm happy to be here. And as a representative of a Russian regulator, I'm really pleased to see all this with my own eyes. Thank you very much.
Is a final stage of a long path. If we can say that the theater starts off by putting away clothes, then the vaccine and medication start by the creation, the development in the right scenario. I would like now to give the word to Stanislav Stakov, who the director of the State Institute, because everything starts off by the right practices, good practices. And he is a director of the State Institute of Medicines and Good Practices of the Russian Ministry in Industry and Trade.
Спасибо. Я хотел бы, во-первых, пользуясь случаем, поблагодарить
I would like to take this opportunity to thank the organizers of this 7th Congress. Dr. Trujin, who is leading the St. Petersburg Institute. This is a scientific research institute for vaccines and serums. Stanislav Uiba, also, who is the general manager of the Meshenkov Institute. I am thankful for the invitation, for letting me participate in this session. I will be a moderator today and tomorrow, and I would like to take this opportunity to invite you to tomorrow's session because it will be very interesting to see how the regulator and the manufacturer interact with each other. From the beginning, this is not the first time that we are interacting with the Institute of Vaccines and Serums, and to see how we are preparing for inspections, for segment inspection, in other words, the regulators, and seeing this interaction of the regulator and the manufacturer, both on Cuba and Nicaragua. It allows us to create a very incredible project in a short amount of time. In 2016, we started off this project, and we did a lot of work which showed the cohesion and the closeness of the experts. And there were many challenges that we have faced, as many of the colleagues have said before me. This was also a challenge for inspectors. We understood that, as anybody, we also had limitations. We had limitations in the inspection because of transportation, because if you can't get to the site, then how can you inspect? So I am thankful to my team and my colleagues who came up with this idea and supported it, because we started to develop it in May to conduct online inspections. Yes, it is a challenge for the inspector to conduct online inspections, because in order to do this, you really need to know the production and the different production areas. In 2020, we conducted over 350 online inspections, and if we look at how we started in 2020 to date, we have conducted over 1,000 online inspections. It is extraordinary and very unique project. And now we are starting to share our experience in this, and we are also willing to share our experiences with our colleagues. And within the framework of the Eurasian Union, we want to use online inspections as part of our activities, and we are also sharing that experience with them. We're also sharing on how the technological advances should be shared, and we have the colleagues from the Nicaraguan Regulatory Authority, and we are more than happy to share knowledge with them. Merkulov has already told us about his experience. We did a very successful fast-track registration from the development all the way to the registration. Before the pandemic, this was unimaginable. But what I think is that all the cases that we have talked about and that we are discussing today, either both here and other international spaces, they allow us to share experiences and allows us to have a close communications between manufacturers, regulators, because we need to look at our relation or look at the relation between the Russian regulator and the Latin American regulator. Again, I would like to thank you for the opportunity to say a few words here. Hopefully, all the decisions made during this Congress will be for the benefit of our countries. We need to focus on the purpose of the regulator. We need to ensure the population an accessible, affordable, quality product. As Victor Trujillo said, we need to merge or have an alignment between the beginning and the end of the process so that we are always on track. Thank you very much. We were talking about the need for pharmacosurveillance when the safety of the product is ensured. Then through pharmacovigilance we evaluate the application. So now we give the floor to the pharmacovigilance entity in El Salvador. So my question is, how does it work in the scenario of a pandemic? Now we will hear it from Medellín de León. Can you hear me?
Okay, muchas gracias.
Thank you very much. Good morning to all of you. I am thankful for giving me the opportunity as the National Medications Institution. We conduct pharmacovigilance, and as other agencies and as the other panelists have mentioned, in the case of El Salvador, we have conducted various fast-track processes. There is a demand for a system with clear guidelines. El Salvador created technical guidelines
pacientes ingresados con COVID-19.
for medicine
Asimismo,
for
desarrolló
patients with
una serie de
COVID-19.
manuales para los
We have a number of
procedimientos de
manuals and
farmacovigilancia
procedures
para las
for
vacunas, y bueno, pues inicialmente
vaccines.
era una vacuna, pero pues fueron incorporándose varias.
We started off with
Todas pues contra
one
el
vaccine, but
SARS-CoV-2
we have
en el Sistema
incorporated
Nacional Integrado de Salud.
more.
En el caso de El Salvador, pues sí creo que la
In the
mayoría
case
de
of El
países,
Salvador,
ha sido el Sistema
in the case of El
Nacional
Salvador,
de Salud quienes han pues realizado el proceso de vacunación, y particularmente en El Salvador creo que fue una interacción interinstitucional que permitió hacer un proceso bastante organizado.
no hay audio en ninguna de las dos cabinas.
Que también permitió
Sí, ahorita se fue, ahorita se fue.
de farmacovigilancia activa, ya que se les podía hacer, bueno, pues
Right now, we have no feed into the English booth.
era posible
We have a
realizar
technical
o mantener
difficulty, and we will sort it out
con,
shortly.
los vacunados en una etapa posterior para justamente hacer esta actividad de farmacovigilancia activa. Bueno, esta serie de lineamientos técnicos, una vez se pusieron en operación,
So then we created the technical guidelines, then created notifications for suspected events attributable to vaccination, and we have about 9,000 or 9,500 notifications that we have analyzed continuously, whether to assess if there's a security threat or a security alert, and we conduct periodic analysis for each type of vaccine. As a regulatory agency, we keep a constant vigilance internationally for each one of the vaccines, at least the ones that we have in use, and also for medicine, not only vaccines but also medications. Of course, vaccination campaigns have increased the exposure to these technologies more extensively. So we keep a continuous monitoring of the activities, of the scientific information that supports the medicine. We are also paying close attention to what other countries are doing in light of these alerts. And perhaps what's more important for us is to understand the epidemiological source and based on which the surveillance or vigilance guidelines were created, and we adapt those to our reality, to our specific reality in El Salvador. In the case of pharmacovigilance, PAHO created a network of focal points for pharmacosurveillance, where we also participate. There's a decent number of countries in Latin America. This allows us to share experiences, share knowledge, and share different approaches that each country applies, and the things that we have in common based on our specific realities. And we also look at the countries that have a more robust pharmacovigilance system. We see what they have identified, or where, in the cases where they have more information about. So this was great because it was a strengthening exercise on how we were doing the evaluation processes, what they were using, what we were using, the periodic security reports, and in general, the pandemic has been a big challenge, but it also helped us create and build more capacity, and especially because of the specific difficulties of the epidemiological conditions which varied along the way through the pandemic. But this flexibilization of criteria in light of the technologies for prophylaxis in this specific case for COVID-19. So we also see the need to stay updated and to create new areas within our institutions and to conduct systematic reviews, because during the pandemic a lot of information was generated and a lot of data to validate authorization process, but also data about the vigilance. So I think we have made good progress, but there are still some challenges ahead, and this has led to improvements in the administration, from the administration standpoint. We've done a lot of the processes either electronically or online, and we also see that we have improved interinstitutional communication, and this has been to overcome all the challenges that the pandemic posed. Thank you very much. Muchas gracias. Thank you very much. Now we have been talking about COVID, right, which has been one of the main issues that we've faced over the past 2 years, and it's been a challenge for the health sector, but also we have other sectors and other institutions like WHO and local organizations that have warned us about other diseases like influenza, and vaccination against other ones is something that we also need to keep in mind. But COVID may decrease, the incidence of COVID may decrease thanks to masks and medicine, but we need to be clear that there are other diseases that also require our attention, demand our attention. So now we have Yasmina Umaña, who is the coordinator, EPI coordinator of the Ministry of Health of Nicaragua, and she will talk about the immunization for influenza. Thank you very much. I would like to thank all of you for the opportunity to participate in this panel, in this 7th Congress. We have made great progress on this specific regard. As background information, I can tell you that Nicaragua is characterized for having a very robust immunization system. We currently have 12 vaccines that are divided based on age, and the population trusts the vaccines, and our government really makes an effort to have vaccines available in the different health posts. It limited the access. We had an issue that limited the access to vaccines. Nicaragua didn't go under quarantine, and Nicaragua is one of the only one of the few countries that didn't install or applied a quarantine. And due to the pandemic, we had to develop a different strategy. The Ministry of Health had as its main strategy to provide vaccination for the population was done through regular vaccination scheme. Due to the pandemic, we had to deploy other strategies. Through our family and community health care model, we were reaching out to the people, to the population, doing home visits and going to work sites and going to schools, meaning beyond the health posts, beyond hospitals. And we took the regular vaccination scheme right to the population, and we got the expected results. I believe that this is an important milestone, and we relied heavily on health workers, our health workers. They were instrumental in this process because they went to the communities and to provide vaccination, and we were able to provide vaccination continuously without interruption throughout the pandemic. Another vaccine that we have included is for children. So now the COVID vaccine is part of our regular vaccination scheme. In other words, we don't say, oh, this is only for COVID, or you only need vaccine for COVID. No, now COVID vaccine is part of the regular vaccination scheme. And we have achieved this thanks to a team effort that starts by political will to approve the vaccination budget. We didn't run out of the vaccine supplies despite flight limitations, and without stopping regular vaccination, and because we mobilized health workers to the communities, to the fields. The success of this strategy was a result of a team effort, but most importantly, the population trusts the vaccines because the vaccines that we are provided are free and are quality vaccines and are being offered in every health unit in the country. Thank you very much. Before moving on to the next speaker, I would like to see a slide, the slide that Marcelo has in his computer, because this is showing how there are no barriers to a global problem like this. So the slide that we're seeing right now, DCMN, which is the Federation of Developing Countries, and they showed that over $9 billion were allocated to develop vaccines, and that was distributed as follows. 5% was allocated to developing countries, and 95% of this number was allocated to developed countries. Also, developing countries are the countries that need more vaccines because their density is higher. This is why we say that our efforts need to be balanced and targeted.
Biopharmaceutical Network, que nos va a hablar de la interacción.
So now we will hear about the interaction, because we talked to our international colleagues. We have some joint projects because we are all part of an international organization. Mr. Sarapagnak, we'll hear you now. Dr. Sarapagnak, are you hearing us? Colleagues.
Hello. hear you very clearly. Am I audible?
Yes, we can hear you.
Okay, just allow me to share the slides. Is the screen visible? Is the screen visible?
Да. Да, я слышу. Right, we can hear you. You can start your presentation.
Yeah, I just want to check. Yeah, okay. I just want to check if my screen is visible.
Yes. We have a presentation on the screen.
Okay, great. Thank you. At the outset, I would like to thank the organizers of this Congress and Meshnikov for giving us this opportunity. I'm Sarang and I'm speaking on behalf of Mr. Sayed for EBPMN, which is Emerging Biopharmaceutical Manufacturers Network. Yeah. So just to give you a brief about the EBPMN, I think the pandemic has made it very clear that there was a concerted effort required from every corner and every stakeholder from the industry. And independent bodies played a very significant role. And one such that is coming up is the EBPMN. Is definitely one of the upcoming I would say a platform from emerging countries, which is coming together for the biopharmaceutical products and initiatives by various manufacturers from this part of the world. As far as the legal structure is concerned, this is registered as a Section 8 company under the laws of India, and it is functioning as a non-for-profit association. The secretariat for EBPMN is based out of Brazil. The mission, of course, is to enhance the access of biopharmaceutical products and manufacturing capacity building in low and middle income countries. The value proposition being, such a global platform is required in the area of biopharmaceutical, more so in the area of monoclonal antibodies and biosimilars on a therapeutic side. From every nook and corner of the industry, including the public sector, private sector, academia, non-government organizations, and other research institutes and organizations, and of course, to mention the WHO and UN healthcare agencies. Currently, we have Sinopharm. As you know, Sinopharm is one of the largest manufacturers of biological products from China. We have BioVac from South Africa as a member. BioVac has been in the industry for donkey's years and has been contributing significantly for the manufacturing of biological products, including vaccines to a great extent in South Africa and southern part of the African continent. We have Instant Butantan from Brazil. We have Tech Invention from India, and Zydus, of course. Zydus being one of the leading manufacturers in India. And as you're aware, they're the only ones who could manage to manufacture a DNA-based vaccine against COVID-19. Zydus is also one of the very few companies who have been able, who has been able to manufacture a cocktail monoclonal antibody against rabies disease. We also have Panacea Biotech as one of the members. Panacea has been the pioneer in vaccines manufacturing in India, and they are the ones manufacturing a lot of vaccines and have a lot of molecules, biological molecules being researched and developed in-house. As an agenda, since 2021, EBPNM has been conducting a lot of seminars, a lot of interactive sessions on various topics, which are definitely important from the perspective of the challenges that are being posed to us. Like the antimicrobial resistance, we need biologics in that area because antimicrobial resistance is the concerned area for everyone. We do make sure at EBPMM that we do take care of this, and we often discuss this issue on a regular basis. To create awareness and also to be able to contribute to address the challenges put forth to us by antimicrobial resistance. That being one area, the discussions have revolved also around a lot of other potential therapies and technologies in biotherapeutics manufacturing, on the regulatory part, on the advocacy with WHO to gain some sort of prominence vis-a-vis vaccines for other biosimilars. On novel biologics and biosimilars is of course, the talent, the cream de la cream from the academia who bring a lot of information to the table. That is insightful webinars we do arrange. We have timely update on guidelines that are being brought up by various countries with respect to biologics, biosimilars. We have been able to initiate few programs which are co-development initiatives and opportunities. So the manufacturers can come together, can pool in the strengths. Maybe one can bring in the research and development part, and the other can bring up the manufacturing part. So even those possibilities are being worked upon. We are working towards attracting funds, grants in the low and middle income countries for this research and development work, as well as the manufacturing of biologics. We are working on networking opportunities across the members from the emerging countries. Apart from this, what it can benefit at a global level, and to take EBPMN to a global platform is a better understanding between emerging biopharmaceutical manufacturers from this region, better understanding and coordination with World Health Organization and other regulatory agencies. We are closely following up and working to bring in NGOs and universities so that, and fresh talent, fresh information comes in to this forum. Other biotech service providers, we are also working together to strengthen the capacity building of manufacturers, biological manufacturers. Last but not the least, we are working together to ensure the access of training programs for the workforce development across the countries and for the members of EBPMN. So this has been our endeavor to take this platform to a global level and to help emerging countries develop the capability of manufacturing biological products in of course, the low and middle income countries, because this is the area which actually requires these products the most. So that is all from my side, ladies and gentlemen. Thank you very much for your patient hearing and audience. Thank you.
Спасибо большое. Спасибо. Уважаемые коллеги, я благодарю всех участников нашей сегодня панели.
I thank all participants for this session. I think we did very fruitful work. Thank everyone for having been with us and for listening to us. Because I think we're still pretty much on time. So I think we now take a break, and thank you all very, very much for your kind attention.
Объявляется перерыв.
And I refuse at the moment to specify for how long.
Muchísimas gracias, compañeros y compañeras, participantes del primer panel de esta mañana. Vamos a pasar todos al Salón Los Lirios. Disponemos de 1 hora para el almuerzo. Muchísimas gracias.
Aló, ¿me escucha, Marcelo? Sí. 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 Ok, yo no lo escucho. Yo te escucho acá. Sí. Ok. Que te haga la traducción entonces. Espérate, me lo estás tirando al canal en español. Me lo estás tirando al canal en español. Se tiene que ir al canal en inglés. Probando. Sí, en español. En Zoom en español lo tengo. Sí. Sí. En español.
Es al TriCaster. Vos tenés que mandarle solamente
el español.
Marcelo, ¿vos me escuchas? Sí. Ok, lo tengo en ruso y en español. Lo tengo en ruso y en español. No, no tiene que ser así.
¿Solo español lo querés?
No, a ver, olvídate que yo hablo español. Yo estoy hablando inglés ahorita.
¿Lo querés?
Lo vas a enviar al canal en inglés y se lo vas a mandar a los traductores.
No, ahí vamos a hacer como todo lo que está todo lo que está en vivo. ¿Hablado?
Probando. Mira, pero yo lo estoy escuchando aquí, en el Zoom de español y en el Zoom de ruso.
En español, ¿verdad?
Probando.
No, en español.
Probando. Sí.
Sí, en el español tienes que escucharlo. En el español tienes que escucharlo porque yo se lo mando a él. Ahí sí tienes que escucharlo. No, a ver, espérame.
Pero es que no deberían, sí, porque si no...
Vos tenés muteado todo ahorita, ¿no? Todo el tricaste le está mandando. Ya le vas a mandar ahorita. Dale. ¿Que hable?
¿Quién? Marce. Habla. Yo no escucho a Marcelo en ningún lado. Verificame que Marcelo tenga bien configurada la pieza esa.
Estaba el Zoom.
¡Aló!
Y estábamos escuchando la cabina de acá. No
Hola, te escucho.
Ya, ¿qué fue lo que les pasó a ellos? Se metía el piso de inglés a ellos. Eso no nos...
¿Probando? Yo lo tengo en los dos canales.
Yo solo te escucho a vos y no escucho a él.
A él no lo escuchas. Pero es que eso es ayer, sí.
Cabina la escucho.
Lo que vos estás hablando.
¿El quién?
En la traducción. Yo le estoy mandando la traducción y él me está mandando otra vez.
¿Probando? Ok. Pero yo me estoy escuchando... ¿Quién me mutió? ¿Probando? Sí. En el español estoy yo ahorita. Yo me estoy escuchando en el español.
El inglés, que el inglés tiene
Mandar audio en inglés.
Sí.
Habla en inglés.
Hi, good morning.
No, habla en el inglés.
Es que estoy hablando en el Zoom. No, el de retorno. Yo me estoy escuchando de retorno por el Zoom en español. O sea, están inyectando en el Zoom en español el audio en inglés. ¿Ya me entiendes? Pues, si yo hablo en inglés, va para adelante.
Bueno, eso me va. No tenemos YouTube, creo. Ok. ¿Qué se llama? ¿Cuál es el audio que yo te mando a escuchar? Sí, solo habla, solo habla.
Sí, sí, sí. 1, 2. Sí, sí. Inglés. 1, 2, 3, 4. 1. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 1, 1, 2, 3. Action. 1, 2, 3. Action.
Y te voy diciendo que habrá las cabinas uno a uno.
Sigo hablando de todo. Sigo hablando. 1, 1, 1.
Llamarlo con
1.
Vaya. Sí.
1. 1.
¿Habla?
1, 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 1,
Que abra la
1.
cabina ¿cuál? ¿El de español? La cabina en español.
1, 1, 1, 1. 1, 1, 1, 2, 3. Action. 1, 2, 3. Action.
No tengo nada todavía.
1, 2, 3.
¿Quién va a hablar? Pero va a hablar en la segunda cabina. Porque la primera cabina es ruso-español. Y estamos suponiendo de que le están inyectando inglés. Es que vosotros tenéis una sola línea. No, ellos te mandan una sola línea o tres. Ok, que habla donde está él, no sé cuál sería.
La segunda cabina que hable.
No, él que
Marcelo.
Que le enciendan aquí en la consola.
Ok. Abrirle. 1, 2.
Inglés. Inglés, inglés, inglés.
1, 2, 3.
Inglés. This is the English channel, testing 1, 2, 3. 1, 2, 3. Pro. Inglés. Inglés. 1, 2, 3. 1, 2, 3. Inglés. Inglés. This is the English channel, testing 1, 2, 3. Listening to original. Original audio. 1, 2, 3. 1, 2, 3.
1, 2, 3. Action. 1, 2, 3.
Este es el canal en inglés, hablando desde la cabina de inglés.
1, 2, 1, 2, 3. Action. 1, 2, 3. 1, 2, 3. Action. 1, 2, 3. Action. 1, 2, 1, 2, 3. Action. 1, 2, 3. Action. 1, 1, 2, 3. Action. 1, 2.
This is the English channel, testing 1, 2, 3. English channel. English, 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 1, 2. This is the English channel, testing. Today we'll have the keynote speech by Sergey Arakelov, PhD and the head of the office.
1, 2, 3, action. 1, 2, 3, action. 1, 2, 3, action. 1,
Chacín, seguí hablando aquí, voy a ver café.
Llevamos horas preguntando que si se puede,
Que se puede
que ellos no traigan café y nada.
Pero ya está.
No jodas.
¿A quién le preguntaron?
A los meseros que estaban afuera. Bueno, ahorita voy a ir.
Siempre que mandamos una para acá, viene también la señal que no debe estar así. Decirle, después de marcar lo que hace.
Pablo. 1, 2, 3, action. 1, 2, 3, action. 1, 2, 1, 1, 1, 1, 2, 3, action. 1, 2, 3. 1, 2, 3, action. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 21, 22, 23, 24, 25, 26, 27, 27. ¿Cuál? La mía.
Voy a ir a avisarles que. Porque ellos son los que querían, yo
te traigo.
Sí, no importa, voy a ir a dejarse.
No, ese está bien. Este retorno.
¿Cuál es el retorno? ¿Cuál es lo que vamos a mandar?
O sea, yo tengo este micrófono.
Sí.
Este es el audio que me están mandando ellos.
Ok, y este está bajito.
Sí, está bajo.
Sí.
Y este es el relay.
¿Y este qué es?
Este es el power que para que se haga la combinación entre esta cabina y la otra.
Ok.
Cuando es relay.
Y los dos hacen la misma cosa.
Sí, los dos.
Es que aquí, y este que es la entrada de audio que tenés allá por ahí.
Esta.
El OR dice aquí. Es esta. O sea, de alguna forma que lo que me está regresando ahí, porque esto mixea también.
¿Cuál es el audífono que traduce ahorita de inglés a español?
Cualquiera de los dos.
Ok, entonces habla.
Yo aquí no estoy escuchando nada en la
Aquí no
cabina.
tenemos nada. Hola, 1, 2, 3, 4. Aquí está este es el inglés.
Ahí escucho suave este. Ahí es bajito. Te voy a ponerle dándole churupaco el volumen.
1, 2, 3, action.
¿Vale?
Hola, 1, 2, 3, 4.
Bajito.
Bajo. Sí.
¿Qué hable?
Dale. 1, 2, 3, 4, 5, 4, 3.
¿Qué hable, va?
Hola.
Ahí lo escucho mejor a él.
1, 2, 3, action. 1, 2, 3, action.
Ahí lo escucho.
1, 2, 3.
Mira, vos mueve estos parámetros. Yo le voy a decir a Iván que te estaba diciendo, mueve estos parámetros de la entrada para ver si eso se modifica. Mira el sonido.
Ok. Hola, 1, 2, 3, 4, 5, 6, 7, 4, 3, 1, 2, 3, 4, 5, 6, 4, 3, 2, 3. Sí.
1, 2, 3, action. 1, 2, 3, action.
Hola. 1, 2, 3, 4, 5, 6, 7, 4, 3. 4, 4, 4, 5.
1, 2, 3, action.
4, 4, 4. 4, 4, 4, 5, 6. ¿Ah?
1, 2, 3, action. 1, 2, 3, action. 1, 2, 3, 1, One, one, two, three, action. One, two, three, action. One, two, three, war. One, two, three, action. 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 4, 5, 6, 7, 8, 9, 10. Iván, yo no te escucho, Iván, 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 no te escucho. Iván, no te escucho.
Está hablando, sí. No le escucho, pero ya el retorno ya se le bajó, ya no le escucho. Ahí sí. Este es.
1, 2, 3, action. 1, 2, 3, action. 1, 2, 3, action. 1, 2, 3, action.
En español. Mira. En español es que estoy hablando ahorita. Hola, 1, 2, 3, 4, 5, 4, 5, 4, 3. Es correcto, ¿qué tal el inglés?
Sí, es que estaba en español.
Hola, 1, 2, 3, 4, 5, 4, 3.
Ahora,
1, 2.
Si vamos a cambiar transmisor, cambiémoslo ya.
Es que no es. Ok. 3, 3, 4, 5. 4, 4, 5, 4, 3. 1, 2, 3, 4, 5, 4, 3. Hola, 1, 2, 3, 4, 4.
No estés llorando después cuando ya te levantes y se te meten.
4, 4, 4. Yo estoy en el canal de inglés. En canal de inglés. Canal de inglés, canal de inglés. Hola, hola, hola. 1, 2, 3, 3, 4. Aquí te lo. ¿Qué pasó? Mira, no se le mete ya el home. No. Mira. Aunque está medio. Por eso. Acuérdate que estéreo no. Sí, okay. Todo es mono. Sí. ¿Pero que no cambia el televisor? No, así déjalo. Ya por más ni lo toquemos.
El audio, el video no lo estamos haciendo.
No, sí lo estoy haciendo.
Iván, necesito que el video que nos manden, que sea el video.
Ya le dije, vení completamente, brother, que es el que sabe.
Hola, uno.
Testing, testing. Testing, testing, testing.
Ahí te escucho bajito. Ok. Abrí la
Testing, testing, testing. We are testing the English language.
Lo escucho a los dos.
Okay. Marcel, ¿escuchas? Marcel. Iván, decile a Marcelo de que si escucha los dos audios, solo el tuyo. Ok, ok. ¿Estamos bien entonces? No, ¿por qué?
Sí, por eso. Yo debería estar escuchando bien a Iván. De la última cabina. Yo
Sí.
escucho a él.
Y a mí.
Y a Iván, pero a Iván lo escucho bajito y a él lo escucho alto. A Marcelo.
Ah, porque no te están enviando el audio de la primera cabina. ¿Pero ahí?
Ahí escucho a Iván. Ahí escucho a Iván nada más. ¿Habla Marcelo?
Marce, habla. Iván, decile a Marcelo que hable.
Yo a Marcelo no lo escucho.
No lo escucho.
Súper alto. Súper alto lo escucho. A él no lo debería escuchar. Es cuando se mixean entre una y otra, porque creo que por la misma línea te vienen las dos ahora. Sí. ¿Escuchar el solo de la cabina de Iván? ¿El solo de la cabina de Iván? De la última cabina. ¿Qué escuchas? ¿Habla Marcelo? ¿Qué estás escuchando vos?
Iván.
Y ahí escuchaste a Marcelo. Okay.
Iván, decile a Marcelo que hable. Está hablando. Iván. Iván, habla.
Ahí lo escucho. Entonces después, cuando vamos a mandar los dos. No, a mí solo mandame, cuando dices tú, deja tu casa, a mí solo mandame la última cabeza.
Habla, Iván.
Que hable Iván. Sí, Iván. Que hable Iván.
A Iván lo miro en su canal y a Marcelo lo miro en su canal. Yo. Hablen los dos.
Yo no tengo ninguno.
El TriCaster no tiene ninguno. ¿Ves?
Debería estar solo el inglés. No está en inglés.
Espérame, ¿habló?
Probando, probando.
A él lo escucho, pero no escucho nada de lo que están hablando. Ellos dos están hablando y no escucho nada.
El TriCaster no está recibiendo el audio de la primera cabina ni de la segunda cabina.
Ahí
tiene que recibir las tres.
¿Habla él?
Habla, Iván.
Los dos. Ahí tengo los dos.
Y a mí también.
Solo la última, ¿no? Aquí ya no tengo
Sí.
nada.
Iván, habla, Iván.
Ahí tengo Iván. Ahí solo tengo Iván.
Ese es el español solo.
Ahí solo tengo Iván. Así debe ser. Y tiene el phone. Mándame a él, solo él a Rusia. Ahí tengo. Ahí solo tengo él. ¿Vos?
Yo escucho los todos. Yo los escucho bien, a cada uno en su canal. El español en español y el ruso.
Lo tengo bien, entonces mándame solo Iván ante nuevo. Ahí está. Así debe ser.
Solo es español. El inglés no me está llegando.
El inglés no se está llegando. Bueno, igual ya no va a haber traducción en inglés.
Pero es que no va a recibir inglés, porque yo estoy hablando inglés.
No,
La
por
primera
eso.
cabina lo va a traducir a español.
Por eso, el que está hablando es en español.
Exacto.
David.
Inglés no vas a recibir.
Okay.
Pero es que tiene que hacer el cambio, ¿sí? Para mandártelo por la otra señal.
Porque hay que arrancar el Zoom español, que vas a conectarse a una señora y preparar la presentación. Creo que las pruebas del inglés podríamos hacer después de que se termine.
Ahorita ya está,
Ya se compuso.
Sí, ya.
¿Qué fue el
Ahí están todas las cosas. Ya aquí no están.
Ok, entonces ahorita ya no tenemos...
Hay que ver restando ahí de cada traslado.
Pero en inglés, cuando lee chino, o sea,
¿Cuándo lo tiene?
mañana va a haber un montón de habladera en inglés. Mañana, 3 habladores en inglés.
O sea, que hoy otra vez hasta las 3 nos
Ellos
quedamos.
sí tienen que cambiar el auricular porque se escucha, pero es el auricular porque lo cambió, sin tocar nada más, solo cambió el auricular y ya suena mejor.
Bueno,
mañana quería probar el canal de inglés también.
Hoy en la noche hasta las 3 de la mañana.
Mañana la primera sesión sería full inglés. Y un poquito de español.
Español e inglés. Y después en la tarde solo hay español. Ahorita tenemos español.
Ahorita
Mañana
tenemos
no tenemos material para pantalla, para mañana, algo específico.
Ahorita hay que terminar eso.
Lo de ahorita en la tarde.
Después voy a comenzar a buscar todo eso.
Si querés ahorita practiquemos el... Puedo hablar por el Zoom en español para simular de que es la transmisión.
O sea, le van a hablar, perdón, en el inglés.
Él va a recibir...
Esto.
Solo uno me trajo este, dice que se le mete, dice este.
¿Con ese se le mete?
Sí, Jon. Ya. Pero vale, hablarles en inglés vos. Este es el otro audífono que tenía David, que no se escuchaba, dice.
Ajá.
Pero ya que se escucha.
No sé porque no estoy escuchando, no hay nada ahorita en el canal. Esta es la cabina de inglés, probando 1, 2, 3, inglés, cabina de inglés. This is the English channel, testing 1, 2, 3. 1, 2, 3, testing English channel from the English booth. This is the 7th International Russia-Nicaragua Congress. Testing English 1, 2, 3. Can you hear me? Sounds loud and clear. Testing 1, 2, 3. 1, 2, 3. 1, 2, 1, 2, 3. Pero, vos decís que era cuando, porque era don David, ¿verdad? Que estaba hablando en ese momento.
Sí, yo cambié el audífono, este me lo prestó tu muchacho.
Correcto, pero fíjate que...
Se escucha bien.
Pero está raro, sí, porque no creo, mira, eso pasaba antes con aquellos, que aquellos son huecos por dentro y pasan, no escucho. Son huecos por dentro y pasa. ¿Vos escuchás? No se escucha. Y lo que escuchás aquí se pasa al micrófono, ¿ya? Como es hueco, una sola cavidad, entonces con aquello. Pero con estos no hay forma, pues, de que esto le transmita al micrófono, ¿ya? Tiene separación, tiene este cable, tiene esto, que es igual que esto. Ahora sí, pero bajón.
Sube en español.
Ahora sí, pero solo en este audífono. Sí.
Zoom en español.
Sí, pero solamente, bueno, sí hay. ¿Vos escuchás bien?
Sí.
Ajá, ahora sí. Ahora sí, es que es como un mal contacto, fíjate que tiene ese.
El audífono.
Okay.
Seguro en algún momento lo tironearon o algo así.
Sí. Ahora escucho, pero quiero escuchar en el tuyo. Que siga hablando en el piso, para escuchar el piso.
En español, Zoom en español.
Ah, este es Zoom. Zoom en español. Sí, este suena mucho mejor.
Zoom en español.
Van a salir a comer.
Zoom en español, Zoom en español.
¿Cómo?
1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. ¿Me escuchan? 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. Mira, ¿quién le está inyectando el audio del Zoom en español?
¿Y ese cómo lo escuchamos?
No hay nada ahorita.
Ahorita no hay nada. Ese es bueno, fíjate, ese
Sí.
no ha dado problema. Y este, ¿sabes? Este aquí, te fijas, este no es hueco como
Sí.
este es hueco, entonces este siempre nos da.
No se mete el...
No es hueco. Y aquí, lo que se escucha aquí, se pasa aquí al micrófono.
Y este mejor, sí.
Ese sí, pero no, lo quitaron.
Yo lo quité porque David estaba traduciendo bien.
Sí.
Lo voy a volver a poner. Pero es que ahora sí, bien.
Escuchas bien, ajá.
Bien. Necesito...
Zoom en español.
¿Habla vos?
1, 2,
Zoom en español.
2, 3, 4, 5, 6, 4, 3. Yo lo escucho bien.
Este es el inglés, este es haciendo la prueba desde la cabina de inglés. 1, 2, 3, probando de la cabina de inglés. Cabina de inglés. Cabina de inglés. Ahora, la prueba real se tiene que hacer ahí, que sigan hablando para que no se metan.
Ahí ya se murió. Se murió todo, sí. Yo no escucho nada.
Hablando.
No, pero es que estoy en inglés, sí, me dio que en inglés.
Ahorita en inglés.
1.
Ah, ok.
Yo te...
Sí,
Sí, yo te escucho. ¿Vos me escuchas ahí? Sí, perfecto.
Sí, me estás escuchando.
Perfecto. Yo ahorita estoy en el canal de inglés.
Zoom en español.
Correcto. Sí. Yo estoy...
Zoom en español. ¿Me escuchas, Iván?
Sí, yo estoy en el canal... Sí.
Porque los muchachos ponen...
Sí, yo estoy en el canal de inglés. Sí, te escucho bien,
Ahora,
te escucho bien.
El otro señor, no sé cómo se llama.
José Daniel.
En la cabina, inclusive no te escucho. No sé por qué no te escucho, porque no se está inyectando los otros. Sí, pero no lo está inyectando, lo que ellos están traduciendo, no lo está inyectando a los Zoom en español. Lo de las cabinas. Una es inglés, lo vamos a mandar al canal en inglés. Y el otro es el ruso. El canal ruso.
Hola.
Hablen los dos.
Ok, yo estoy en el canal de inglés. Yo estoy en el canal de inglés. Cabina de inglés. 1, 2, 3, 4, 5, 4, 3, 4, 1.
Habla.
Ok, yo estoy en el cabina de inglés. De inglés. Hola. 1, 2, 3, 4.
El audio que ustedes están traduciendo, yo no lo estoy escuchando en ningún Zoom.
Es el piso.
Entonces, hablen para que lo identifiquen ahí.
1, 2, 3, 4, 5, 4.
No tengo nada.
4.
No tengo ninguno del audio de las dos cabinas.
Hola, 1, 2, 3, 4, 5, 4.
Ustedes ya verificaron
1,
que
2,
están
3,
traduciendo
1, 2, 3.
bien, con
Sí.
la maquinita, ¿verdad?
Claro.
Sí, está bien.
Sí.
Ellos están hablando, yo no los
Hola, 1, 2, 3, 4. Yo estoy en el cabina de inglés. Yo estoy en la cabina.
Ya te escucho.
Estoy en la cabina de inglés.
Está en el canal inglés.
Inglés.
En el inglés.
Yo estoy en cabina de inglés.
Sí. Mira, ahorita.
Hola, 1, 2, 3, 4, 5, 4, 5. Ok, 1, 2, 3.
Y está en canal inglés.
Estoy en el canal de inglés.
Pero no está bien. Es que él va a escuchar de mi español y lo va a traducir en inglés.
En inglés.
Y lo va a inyectar en el canal inglés. Está bien. Ese ya no se mueve. Ese ya está ahí, está bien. Le falta el otro ahora.
Ahora.
El otro es el ruso.
El ruso.
Escucha mi español y lo traduce a ruso.
José Daniel. José Daniel.
Habla.
Probando.
Ahí está, lo está inyectando en el canal ruso.
¿Qué nota?
Ok, 1, 2, 3. Ahí está en la cabina. Ahí está en la cabina. Yo.
Ajá.
Ahí estamos bien.
Aló.
¿Quién habla?
Ruso se escucha. Bárbaro.
Aló. Ahí estamos entonces. Eso es lo que viene ahorita. Sí.
Probando ruso, 1, 2, 3, ruso.
Ay, voy a abrir la otra.
Ruso. Ruso. Probando el canal de ruso. Canal de ruso.
Mierda. ¿Qué es esto? A ver. Esto es español.
De la vieja,
Es la primera. Este es español. Calma, no me perdas ahorita. Español. Es esta.
Probando canal inglés. Ya.
Pilar, buenas tardes. ¿Me escucha?
Hola, buenas tardes.
Buenas tardes. Como media hora atrás se acabó la primera sesión del panel. Ahorita todo el mundo está haciendo su almuerzo, entonces hay que esperar tal vez una media hora o unos, sí, más o menos. Y comienza la segunda sesión para que no se...
Entonces, más o menos a las 2 empezaría.
Sí, más o menos a las 2, correcto.
Ok, perfecto. Muchísimas gracias. Voy a estar conectada aquí por cualquier cosa, entonces.
Ok, perfecto. Correcto, así es.
Gracias.
Muchas gracias.
Sí.
Buenas tardes, invitamos a todos a pasar al salón.
Дорогие друзья, проходите на свои места, и мы начинаем работать по второй части нашей программы. Пожалуйста, занимайте свои места, присаживайтесь. Большое спасибо.
Внимание, прошу занять места. Мы начинаем.
Por favor, tomen sus asientos y empezamos.
Пожалуйста, займите свои места, мы начинаем наше заседание. Уважаемые товарищи, дамы и господа, приветствую вас.
Посвященную биофармацевтике будущего «Новые герои. Подготовка кадров для послезавтра». Сегодня мы с вами поговорим о успешно применяемой формуле нами «Наука плюс образование плюс индустриальный партнер». И мне уже не терпится перейти к событиям, важным событиям, которые сейчас произойдут в рамках нашей панели. Я бы хотела пригласить Виктора Палыча и доктора Рамону для подписания нашей программы «Бионика», чтобы в дальнейшем передать ее в Министерство образования. Прошу вас, доктор и доктора. Прошу вас подписать и обменяться папочкой. Мы подписываем нашу обучающую программу на 2 языках, на русском и испанском. Muchas gracias.
Спасибо.
Прошу вас, коллеги, продемонстрируйте нашу обучающую программу, подписанную и утвержденную между 2 учреждениями, нашей компанией и университетом. Это наш 2-й официальный документ. И теперь перехожу к не менее приятному событию. Виктор Палыч сегодня вручает 2-ю книгу в рамках нашего проекта «Бионика». Книжка-комикс, которая написана нашими сотрудниками, наших компаний на понятном и доступном языке, потому что знания должны быть не только полными, но еще и интересными. Прошу вас.
Ну, доктор Елена не дала мне возможности ничего сказать про книжку, все уже рассказала. Да. И действительно, согласно той доброй традиции, которую мы будем стараться выдерживать, в прошлом году мы разработали, выполнили и привезли 1-й учебник. Ну, скажем так, он достаточно интересный, да, и он положил основу дальнейшей работе, нашей работе над этой литературой. И я так понимаю, что студенты, которые обучаются в Российской Федерации, они со следующего года точно так же уже вполне себе профессионально приступят к подготовке следующего обучающего материала. Этот вариант очень необычный. Этот вариант очень необычный, очень интересный, очень такой интерактивный, но во всех смыслах узнаваемый. И те, кто будут работать с этой книгой, те, кто будут работать с теми людьми, о которых здесь идет речь, конечно, это будет очень приятно и очень комфортно. Он такой персонализированный. Доктора?
Por favor, tomen asientos, estimados colegas. Y ahora voy a presentar la ponencia para a continuación pasar a la sesión de preguntas y respuestas que vamos a hacer a nuestros expertos, líderes de opinión que están presentes hoy en el presidio. Continuando con lo del libro, yo voy a añadir lo siguiente. Al leer este libro de cómics, van a encontrar a todos los personajes, van a encontrar como personajes a los presentes hoy que ven en el presidio. Los conocen a todos, a Víctor Pavlovich Trujín, a Stanislav Uiva y a los demás que participaron en este proyecto de Bionica. Y ahora vamos a pasar a las ponencias. Por favor, pongan la presentación en la pantalla. En mi ponencia vamos a hablar de la integración de las universidades y el empleador, de los resultados de la interacción de nuestro modelo entre el Instituto Mechnikov y Instituto de San Petersburgo y UNAM Managua. Vamos a presentar nuestra experiencia. Les voy a mostrar nuestro modelo de interacción. ¿Cuáles son los papeles dentro de este modelo? Todo inicia en UNAM Managua, que es el donante que nos facilita el potencial, que son los estudiantes que llegan al proyecto Bionica. Después trabajamos con este potencial, o sea, con los estudiantes. Y estos chicos, a los mejores, los enviamos a estudiar a la Federación de Rusia. Ahora vamos trabajando con dos universidades rusas y estamos negociando con la tercera universidad para también conectarlos a este programa. Sería la Universidad Federal de Ural, de los Urales. A continuación, en nuestro modelo, como pueden ver, están dos empleadores. Es el Instituto Mechnikov y el Instituto de Vacunas y Sueros. De hecho, es la misma empresa. El Instituto de San Petersburgo asegura las prácticas, la capacitación y la financiación en forma de becas. Y al final, los estudiantes ya bien preparados con diplomas rusos llegan a colocarse para trabajar en el Instituto Mechnikov. En la siguiente lámina ven los resultados de este modelo.
Мы имеем на сегодняшний момент, и это всего лишь за чуть больше года. За 2021 год мы имеем 3 студентов, из которых один это у нас аспирант, и этот и следующий год он обучается, и, можно сказать, через 1,5 года он уже вернется в Никарагуа полноценным студентом с кандидатской степенью. И также мы имеем 2 студентов, которые обучаются на специалитете в Санкт-Pетербурге, в Санкт-Петербургском химико-фармацевтическом университете.
Que estudian en San Petersburgo. Otro estudiante estudia en la Universidad de Tomsk, Universidad Politécnica de Tomsk, en el programa de grado o de bachillerato. Ha pasado un año desde el congreso anterior y ya tenemos un contrato suscrito entre la Universidad de Managua y el Instituto de Vacunas y Sueros. Así que ya podemos hablar de ciertos resultados. Los resultados de trabajo de estos 2 años se manifiestan en nuestros libros de textos, cuyos autores son empleados del Instituto Mechnikov y Instituto de Vacunas y Sueros de San Petersburgo. También tenemos como coautores a los profesores de la Universidad UNAM Managua. En esta lámina yo quisiera comentar un poco sobre estos chicos. Son 2 estudiantes de UNAM León y 2 de UNAM Managua. son embajadores de, así, para así llamarlo, de nuestro so to speak, and they are studying in Russia very enthusiastically. I would also like to show you on the last slide that on the 1st of November we will start the Bionica 22 project, during which we will select 10 students, 7 of the UNAN in Managua, 1 from León, and 2 others of 2 different universities, and then we will see who of those will be selected to go to Russia and study in the Russian universities. Thank you very much for your attention. I would now like to give the floor to the speaker of the next presenter, Sara Nagarish. Please, the floor is yours. It is a pleasure to share with you It is a pleasure to have you here and to be able to share the experiences that we have had with the St. Petersburg Institute as well as the Latin American Institute of Technology in Mexico. I will now share with you how the curriculum of physics and chemistry of the faculty and study plan has changed on the basis of this joint experience. It is important to mention that the major in chemistry, pharmaceutical chemistry, as in the case of other majors, has been changing over the last years, over the last 20 years. This transformation has to go hand in hand with consultations and participation of private sector companies as well as other national and international entities and organizations in charge of guaranteeing and producing quality drugs and for the population. On this slide you can see the focus of the major in pharmaceutical chemistry in Latin America. As you can see, this 10 years ago there was a focus on community pharmaceutics and hospital pharmaceutics as well as public health, and there are some topics that don't coincide with the majors in other American countries, as for example clinical laboratories, food and chemistry. We would like to share with you some experiences of specific countries. On the next slide you can observe that the countries that are at the vanguard of the development of the major on biotechnology are basically Argentina, United States and Colombia. Up to now, I think that in current times many Latin American countries have included in their study plan the teaching of biotechnology and similar sciences, because we have to remember that biotechnology is a discipline that feeds on other disciplines such as biology, organic chemistry, and even informatics or computer sciences. On the next slide you can observe how the curriculum has developed. Before 2008, these were the strong areas of the major: hospital pharmaceutics, community pharmaceutics, industry pharmaceutics. And then you can also observe what we have today. We have incorporated other disciplines and
that have increased capacities and competences so that our students can opt to work in biotechnology industries. We have strengthened and included the biotechnology area as well as microbiology and immunology and microbiology. On this slide, I would like to highlight that to the degree that the biotechnology industry is advancing, there are more and more consumers and users of biotechnological products, and we have about 350 million persons that consume biotechnology products, 325 million people, and then there's a considerable percentage for cancer related diseases as well as with transmittable diseases, deficiencies of the immune system, and neurologic diseases. I would like to highlight that we have a map of what areas have developed most so that we may know what lines of research we should strengthen in the future. I would like to say that at present teaching biotechnology in our classrooms has focused basically on molecular biotechnology or biology. We speak a lot about reproduction, transcription of DNA, and the system of genetic expression. We also deal and study recombinant DNA technology. We would like to deepen through this alliance that we have forged today with experts that we're talking about very important topics. We would like to include into this study plan other topics that go beyond. For example, biocatalyzers, interferon systems, and others, to deepen on all the forms of all kinds of technology for these modern production systems. As to this new biotechnology, we would like to orient it or aim it in future more on cell culture techniques, animal cell culture techniques, and others. It is important to highlight that the construction or creation of a curriculum based for pharmaceutical chemistry is normally based on the needs and demands of a society and the growth of the pharmaceutical industry, the national as well as international industry. So these needs are then analyzed at the local, regional, national, and international level, as well as the services that should be offered that would define the products we need to offer. And of course, this will lead us to define the competences that any degree or any professional studying or any student that completes his studies should be able to present. We have documented all aspects of the preparation of this profession, and today we have an alliance with these two institutes, which has given us the possibility to enlarge our vision and advance towards this technical world-class development and knowledge, as well as cooperation with other national organizations such as the National Health Ministry. And these alliances are vital for our students to make their professional practices and internships in order to learn firsthand from experts in the field. In addition, we would like to invite our experts and the international experts to participate more in conferences, for example, and giving short courses in order to give trainings to teachers as well as students. In general, I would like to tell you that we all work for a society, for our society, and so there should be a social impact in form of products, services, and processes of technological nature. And this is possible in the case of our pharmaceutical industry through research and development. Now, as to the Bionica program, it is a flagship project for Managua, and I think for St. Petersburg, for the St. Petersburg Institute as well as for Mechnikov. And as my colleague already pointed out very correctly, we have been working out study materials, academic exchange, encounters have taken place, and we feel very proud to have 4 students that have earned scholarships, and we think they will make great contributions to the development of the enterprise of the company as well as the country as a whole. Thank you very much.
Поблагодарим Сару за такой удивительный и прекрасный доклад по опыту и подходам в обучении фармацевтической химии. И еще раз поблагодарим за то, что Сара подчеркнула, как важно университетам выстраивать диалог с индустриальными партнерами и компаниями. И в связи с этим у меня сейчас есть вопросы к нашим экспертам. И первый вопрос я хочу задать Виктору Павловичу. Виктор Павлович, вы взяли на себя нелегкую задачу, оказываете поддержку не только моральную, но и финансовую студентам, которые едут за образованием в Российскую Федерацию. Какова же все же основная задача взаимодействия ваших компаний, Мечникова, СПБ НИИС и университетов в этом направлении?
Еще раз добрый день всем присутствующим, всем, у кого хватило силы и воли остаться на вторую сессию нашего Конгресса. Елена, этот опыт, он ведь и для нас на самом деле первоначальный. Подобным конструированием обучения, поддержки, создания социального уюта. Раньше мы никогда не занимались, и, само собой, каждый новый подход, каждый Новый год для нас немножко необычный, причем также обучателен. Мы сами в данном случае тренируемся и получаем своеобразный опыт, и, соответственно, опыт планирования будущей траектории. Конечно, каждый человек, начиная с первой группы, которая прибыла, он, безусловно, и уникален, безусловно, общаясь с каждым из них, и эксперты, которые их готовят, и в дальнейшем уже непосредственно я стараюсь для себя представить, кто бы из них в какой части и области был максимально реализован. Я не говорю полезен, да, хотя полезность общества — это еще один из ключевых критериев воспитания человека. Здесь ориентир на реализацию, на что он будет готов. Ведь мы должны с вами понимать, что из каждого из них мы не сделаем глобального ученого. Да? Для этого надо, наверное, еще больше будет учиться, чем даже пройдя университетскую школу в Никарагуа, пройдя уже следующий этап подготовки в разных степенях обучения в Российской Федерации, и получив уже больший заряд, большую потенцию, может быть, шагнуть на более высокую степень обучения, уйти учиться дальше, чтобы получить возможность принимать участие в разработках биотехнологических. Это интереснейшая вещь, это просто потрясающий мир биотехнологий. Но для этого надо будет готовиться. Безусловно, кто-то из них понадобится непосредственно производству. Почему? Потому что в производство должны приходить люди профессионально ориентированные и понимающие, что происходит с теми процессами, которыми они будут управлять, создавать, и каким-то образом добиваться результата. Не каким-то, а вполне себе обоснованным, целевым. И, как говорили сегодня наши регуляторы, выполняя все требования, все эти цели и задачи, которые стоят в производственной цепочке. А кто-то проявит талант преподавателей и вернуться в университеты. И моя коллега-ректор, с которой мы ежегодно согласовываем и обучение, и направления, и интересы, точно так же, я думаю, с удовольствием примет специалистов, которые уже идут с дополнительной гранью, с дополнительным багажом знаний и умений. Они, безусловно, будут важны для будущих обучений. Блестящий доклад Сары с показаниями, как изменилась система подготовки. Я, честно говоря, смотрю и думаю, как-то я тоже упустил этот момент, но такой специалитет очень важен. Это просто на руку всей программе, которой мы занимаемся. Меня сегодня посол Российской Федерации Александр Хохоликов спросил, а что так мало студентов? И, я говорю, господин посол, я в состоянии вытянуть до 10 человек в год. И это интересно будет и программе, и, собственно говоря, подготовке кадров и обучению каждого человека. И, соответственно, я назвал там 3 направления возможного приложения сил этих людей. Кто-то, может быть, уйдет в регуляторику, кто-то уйдет в Министерство здравоохранения, кто-то в прикладном смысле, получив эту дополнительную подготовку, что-то сменит и уйдет в пищевую промышленность, сельское хозяйство. То есть векторов достаточно много. Я бы хотел, конечно, чтобы они все так или иначе находились на орбите мне понятной коллеги Станиславу. И учитывая тот акцент, который сегодня в приветственном слове я уже озвучивал, и, само собой, не единый же согласовывал с правительством Республики Никарагуа и с правительством Российской Федерации, что да, компания Мечников будет расти, и мы часть разработки усиленно переносим сейчас на институт Мечников. Это очень интересно. Разработка должна переходить, соответственно, в доклинические и клинические исследования. И по доклиническим исследованиям мы программу представим университету и будем ее согласовывать, потому что самим ее вытянуть невозможно. Здесь должна быть очень хорошая кооперация и доброе понимательное партнерство в этой работе, в доклинических исследованиях. И, конечно, клиника и, как итог, производство. Я не зря сегодня назвал цифры достаточно большие в своем приветственном слове. Я имею в виду цифры затрат на развитие института вакцины сывороток в Санкт-Петербурге. И еще раз повторюсь и отмечу, что, конечно, подобная проекция, она будет сооружена и выполнена в Республике Никарагуа. Я сейчас буду вынужден чуть раньше уехать, мы со Станиславом убудем. У нас рабочее совещание с министром Лопесом на точке будущего развития института Мечников. Это уже производственные каскады. И, конечно, я заинтересован, чтобы наши студенты, которых мы, сейчас 4 человека, это еще не выборка. Это понятно, что статистически трудно определить, кто из них в каком направлении будет дальше прогрессировать и развиваться. Но во всех смыслах я заинтересован, чтобы вот эти будущие точки развития, чтобы туда шли обкатанные студенты сначала высшей школой Республики Никарагуа, затем теми дивизионами, подразделениями, которые были названы, Лена, в твоем докладе, в Российской Федерации. Безусловно, это расширится. И, конечно, я прекрасно понимаю, что нам надо довести, но я думаю, коллега из Россотрудничества меня поддержит, довести нашу квоту в российском правительстве хотя бы до 10 человек в год. СПБ НИРС это потянет, это не проблема. Осталось с правительством России договориться. И тем самым мы с вами создаем вот именно то сообщество, тот континуум, если так можно сказать, который будет и понятен, и будет развиваться, и уже дальше запустятся механизмы саморазвития. Да. Вот здесь был доклад Севастьяна Рабдано, и многие удивились, почему он, кандидат физико-математических наук, занимается биологией. Да это, на самом деле, все просто. Да. Человек, имеющий фундаментальное физико-математическое образование, был легко переключен на биологию, да, со своим подходом, со своим умением, со своим знанием, да, и с желанием добиваться результата. А когда результат начинает быть ощутимым, и вот тут, знаете, пандемия, как говорится, кому война, кому мать родна, но только во время пандемии мы смогли все результаты материализовать. От идеи до непосредственно зарегистрированного препарата, обладающего всем комплексом потенций и необходимых качеств, которые требуют на то российская и международная регуляторики. Поэтому судьба каждого нам будет важна. И каждому мы зададим должный и нужный вектор развития. И вот уже дальнейшие каскады событий на десятилетие нашего Конгресса, я думаю, тут у нас уже будет солидный отряд студентов, прошедших школу обучения и в Никарагуа, и в Российской Федерации, и, может быть, где-то еще, то есть там горизонты можно расширять дополнительно и до бесконечности. И сегодня выступали парни из биотехнологии индийской. Это потрясные просто технологические комплексы, и мы много работаем с ними вместе с моими коллегами из Института иммунологии. То есть нету предела границам, которые сдерживали бы развитие. Важно, еще раз повторюсь, чтобы программа бионика, о которой мы сегодня много говорим и говорим всегда, это показательная программа становления, именно становления и определения человеческой силы, человеческого вектора. Вот она позволяет сконцентрировать каждого из десятков, я надеюсь, это будут десятки молодых людей, и дать им право выбрать ту волю, те возможности, какие мы им предоставим. А мы их предоставим. В данном случае у нас уже немного вот такая утилитарная задача создать помочь этим людям создать себя и предоставить возможности для их реализации. Спасибо.
Курского института вакцины и сывороток. Следующий вопрос я бы хотела задать Станиславу, генеральному директору Биотехнологического института латиноамериканского Мечников. Станислав, у меня вопрос к тебе такой. Как к работодателю, прибывшему из Российской Федерации и возглавившему такой необычный институт на территории Республики Никарагуа, не только институт, но и завод, и как же университету и бизнесу найти друг друга?
Bueno, buenas tardes a todos. Voy a hablar un poco menos en detalle, más general sobre el tema de formación, de fortalecimiento y desarrollo de talento humano. Y bueno, a veces me preguntan cuántos alumnos del programa Bionica ya tiene trabajando como trabajadores activos en Mechnikov, y hay dos respuestas, ¿ya? Dos opciones de respuesta. Formalmente son 11, ¿ya? Los que formalmente participaron en las sesiones de Bionica y actualmente trabajan con nosotros. 11 alumnos de Bionica. Pero la otra opción, la otra respuesta es que todos 194 trabajadores de Mechnikov son de Bionica. ¿Por qué? Porque nosotros decimos que ya Bionica se nació en 2018. Lo que pasó en 2018 es que nosotros ya ponemos la placa con el nombre de Bionica, porque todos los métodos, el formato de formación, de acompañamiento y fortalecimiento de talento humano ya se cristalizó con el mismo proyecto de transferencias de tecnología y de construcción de la planta del Instituto Mechnikov aquí en Nicaragua. En 2016 hemos contratado el primer grupo de 26 personas. Y con ese grupo ya elaboramos todos los enfoques que usamos hasta la fecha. ¿Ya? Entonces todos son de Bionica. Y claro que este tipo de proyecto como Instituto Mechnikov se requiere un soporte de recursos humanos porque estamos creciendo, creo yo, bastante agresivo. En 2016, 26 personas, 17, 60, 18, 95, 19, el año cuando se lanzó la producción, 120. Ya, entonces en 4 años ya crecimos por 5 veces. Y ahora, como mencioné, 194, casi 200. El próximo año vamos a pasar 200 personas. Claro que este crecimiento tiene que ser acompañado con colaboración con las universidades, con cuerpo académico, cuerpo universitario. Y bueno, lo único que se requiere es la voluntad de hacerlo. Ya, y bueno, en este caso, doctora Elena de verdad está demostrando cada vez ya esta voluntad de hacer cosas baratas, casi gratis. Ya, entonces está creciendo realmente un milagro de nada, de cero. ¿Ya? Y bueno, es la voluntad de cooperar, de intercambiarse de las ideas, de sus visiones, ¿verdad? Porque siempre, cada institución tiene su visión. Ya cuando nosotros decimos que, por ejemplo, ya hemos hablado en 2017 que los alumnos de la UNAM Managua, por ejemplo, bueno, cuando llegan para trabajar ya no tienen una idea clara de qué hacer. Ya tenemos que capacitarlos nuevamente. A veces son capacitaciones de meses. Pero realmente es por eso, ya reducir el periodo de inducción, el periodo de ingreso. Ya que una persona llega ya preparada, entendiendo de lo que tiene que hacer, ya, y lo más importante también modo de pensamiento, siempre la cosa para mí más importante, que la persona tiene esta voluntad o tal vez necesidad innata interna de aprenderse constantemente, de controlar sus actividades, de pensar cómo puede mejorar los procesos, que siempre acepta y considera y evalúa todas las sugerencias y consideraciones de los colegas internos, colegas externos, ya tiene que aplicar y adaptarse y aplicar todas las sugerencias de la inspectoría, por ejemplo, ya, en un proceso normal. Entonces, ese modo de pensamiento también ayuda bastante, que la persona llega preparada. Entonces, es sobre eso. Y bueno, como demostró en una de las láminas, Sara, que han doblado en 10 años, han doblado la cantidad de carreras. Eso es también importante porque cada carrera tiene su bien específica. Hay que poner las personas en lugares correctos. ¿Ya? Es un enfoque bien eficaz que la persona tiene que estar donde quiere que estar. ¿Ya? Entonces, es así, es la vida realmente, muy viva, muy vivo este proceso. Todo lo que está relacionado con recursos humanos, con la gente en general. Y bueno, así funciona. ¿Ya? Claro que en este caso uno puede lograr mucho en periodo corto, ya, 1 año, 2 años por lo menos se requiere, ya, pero las ganancias de estos proyectos son enormes. Porque recursos humanos es de verdad la parte que tiene lo más valor en cualquier empresa. Entonces, aquí estamos desarrollando los enfoques de Bionica desde 2016. Ya en 18, junto con Elena, ya inventamos este nombre, Bionica, que es biotecnología nicaragüense, si alguien no lo sabe. Y bueno, aquí estamos en 2022 con nuevos retos ya, y bueno, con muchos logros, pero además mucho más además. Gracias.
И я извиняюсь, но мы вынуждены отпустить Виктора Павловича и Станислава для дальнейших встреч в рамках нашего конгресса. Спасибо вам огромное. И, Станислав, до встречи 1 ноября. Следующая сессия Бионика. Мы у тебя в гостях. Спасибо большое. Спасибо, коллеги. Ну, а мы сейчас продолжим уже не спеша нашу панельную сессию, наши интересные истории. И следующий вопрос я бы хотела задать нашему прекрасному ректору, нашему самому важному человеку, доктору Рамоне. Вопрос у меня будет следующим. Что дало Унан Манаго взаимодействие с Мечниковым и СПБ НИИС? Что изменилось? Можете нам рассказать? Очень интересно послушать ваше мнение, доктор Рамона.
To Mechnikov and the St. Petersburg Institute, and what has changed for the university, and what has this cooperation brought you?
Buenas tardes a todas y todos. Es un gusto para mí estar participando en este momento en el panel y siguiendo ya los comentarios que me han antecedido de las personas que han participado. Yo considero que uno de los aspectos más importantes para la UNAM Managua ha sido el fortalecimiento de capacidades científicas y técnicas del personal docente, tanto para desarrollar los aspectos de formación de los profesionales en las carreras de química farmacéutica, como también las competencias para prepararnos para la investigación. Esto se ha venido logrando porque hemos estado vinculados desde el inicio del proyecto de vacunas. También se ha tuvimos la oportunidad de que un grupo de docentes pudiera visitar el Instituto de Sueros y Vacunas de San Petersburgo, y eso pues abre una visión de cómo se deben de hacer los procesos de formación de profesionales. Luego también la participación en los coloquios, los distintos coloquios que se vinieron desarrollando para hacer un recuento de cómo estábamos preparados en Nicaragua y en nuestras universidades para incursionar en todos estos temas de biotecnología y cuáles eran las capacidades que teníamos instaladas. También ha sido muy importante el entrenamiento que se ha recibido para aplicar las buenas prácticas en todo el ciclo, práctica durante todo el ciclo de los medicamentos. También es importante destacar que el hecho de que se estén realizando conferencias con científicos de las universidades rusas, pero también de la Federación Rusa, eso nos ha permitido a nosotros aprender, actualizar, prepararse a los académicos que ya no solamente son del UNAM Managua, sino que también de universidades del CNU y también de universidades privadas. Y un aspecto también importante a destacar es el hecho de estar participando en el proyecto de Bionica, porque eso permite darle ya una formación a los jóvenes desde que están en su bachillerato, los que ya están por bachillerarse, para que valoren esa importancia del estudio de la biotecnología y se vayan también fortaleciendo sus capacidades para una vez que ingresan a la universidad. Todo este proceso nos ha llevado a nosotros a hacer también transformaciones que ya Sarita las señalaba, como hemos venido analizando toda la conformación de la carrera de química farmacéutica, qué vacíos tenía, cómo lograr fortalecer las competencias con la incorporación de nuevos ejes temáticos que ya también ella de alguna manera los mencionaba, como es el fortalecimiento de la biotecnología, de las regulaciones farmacéuticas, de la biología molecular, de la microbiología. O sea, identificar cuáles eran las oportunidades de mejora para darle las competencias a los estudiantes, a los futuros profesionales, y que ellos también vayan encontrando vayan visualizando, vayan proyectándose en este nuevo contexto que tenemos en Nicaragua, pero todo esto lo venimos haciendo porque tenemos esta relación de colaboración con el
relationship of the St. Petersburg Institute as well as the Biotechnology Institute. And also we have been now able to see how we're doing with our postgraduate work. And that has been important because in all these years of collaboration we were able to structure a specialty in pharmaceutical regulations, and this has positioned us here in Central America. And as Sara, did research as to where we are at compared to other Central American countries in terms of postgraduate studies, in terms of pharmaceutical studies, and we realized we were quite weak. And so we really need to strengthen this. We will be graduating people with majors in that career and then move on to postgraduate studies. But it's been very important because we've been able to work now in an alliance with the National Directorate of Medicines in El Salvador to develop this program, and we're going to implement it for other also regulating agencies in Central America. Right now we're going through a consultation process through the different universities in Central America, as well as the regulating authorities in the Central American countries with a company from the Pan American Health Organization. And so we've been strengthening capacities and so what's going on here in Nicaragua is now also expanding to the rest of Central America. We'll be working together on that. And this collaboration is a very inclusive relationship because we at UNAN Managua have broad-based participation from the Faculty of Medical Sciences, the Engineering Sciences, which is where pharmaceutical chemistry is in, the Polytechnical Health Center, but also other universities participate that are members of the National Council of Universities, as I said earlier, as do private universities, and this allows also for a close relationship with the state, with the Ministry of Health, the strengthening of relations between academe and companies or the private sector. And this helps us to identify, what strengths we have, but what we also have to work in. And the fact that we have students in the Russian Federation who are studying there, as we have heard earlier, that will make us stronger. It will strengthen Nicaraguan universities because they come back as graduates with perspective of working at the Meshnikov company, but they will also potentially be professors at our university. And we need to also value the Bionica project, because that will provide more opportunities for young people to participate and have then this ongoing education of professionals to strengthen Nicaraguan capacities to carry out research, clinical studies, and all the different assays that are necessary for the production of vaccines and everything else that is generated from the biotechnological industry. So we need to train professionals, strengthen academe, and ensure that students take advantage of this opportunity. And we're deeply grateful to the St. Petersburg Institute and to the Russian Embassy, which is stimulating this academic opening, and also Mexico, the University of Mexico, where we have students in internships, students that study electronics systems, informatics systems, industrial engineering, because since the company is very complex, all these aspects are important as well, and they need to be in contact with different processes, updated state-of-the-art technology, and so on. And I think that will be terribly helpful to them. So thank you very, very much for this accompaniment to strengthen our capacity and position ourselves also not just personally, but also as a country.
Dr. Ramona Rodriguez, Dean of the Yunnan Managua University. In fact, we are now very thankful to Dr. Ramona, because she is giving us so many opportunities for cooperation, and effectively it is very important and a very good idea to make exchanges between students and the Mechnikov Institute in order to practice in different specialties. In the project we signed today, we also have foreseen activities to give students the chance to cooperate with the Mechnikov Institute. In the past, we were more focused on the pharmaceutical aspects and the regulatory framework. Now, we are now broadening step by step our framework for cooperation. UNAM Managua can be considered as the most important donor of professionals for our institute. We are here together with Sofia Hargif of the agency Rosotrudnichestvo in Nicaragua. She will speak to us and answer questions after the presentation by Dr. Ramona Rodriguez. Sofia, I welcome you, and let's see how we can work this out with the microphone. Obviously, Sofia is helping us a lot through her agency with the Russian program, and the students have now already traveled to Russia. Sofia, the question is, what are the perspectives for 2023 for Russian education in Russia for students and for all of us? Mr. Trujin already spoke of 10 students, but I don't know whether this number is correct. I would like to start by explaining what is this program. The program of scholarships of the Russian government is a program that is implemented through the Ministry of Education and the Rosotrudnichestvo agency. It is implemented almost all over the world. We assign quota to almost all countries in the world, quotas for scholarships per country, and we have a website, www.educationrussia.com, which opens calls for candidates from countries to register and upload documents that are necessary for the first phase. Then these documents are evaluated, and maybe it is necessary to take some exams. We elect a list of successful candidates, and these candidates are submitted to quota for students and can enter into these quota to study in Russia. All this process will be something we will present to the University UNAN Managua on the 31st of this month, so that students know what documents we're speaking of. Now, there's not exactly a problem, but an important detail, because this scholarship is not a complete or full scholarship. It is a scholarship for free of cost study, and if the student needs to learn Russian, they have a chance to learn Russian for 1 year. But this scholarship does not cover other costs. Now, Russia is quite expensive for foreign students, and the Russian government allows foreign students to access to Russian universities free of cost. And this selection process that we are making through our website is the mechanism. Now, students that are proposed by the Mechnikov Institute, as the experts have already mentioned, they receive an extra scholarship from the Institute that does not only help them to survive, but to live a little more comfortable, more protected, and they get more orientation in Russia, which is very valuable. But at present, we don't make any preferences. We don't prefer Mechnikov candidates to others, but all candidates that get registered underlie the same conditions. They are evaluated very honestly, and our forecast is that we want to create a cooperation between the Ministry of Education and Russia that will approve everything in order to give Mechnikov the possibility to offer special scholarships so that their students will be out of the ranking process. They will have a special treatment with 4, 5, or 6 scholarships per year, and maybe this number can grow in the future, and they will be the Mechnikov students, the Mechnikov scholarships, and they will be able to evaluate their successful candidates according to their own standards and parameters. And we are working on all of this. This year it might not yet be possible because the process is taking some time actually, but we are working on it. So now the candidates that are proposed by Mechnikov will participate at the same level as other proposals from Nicaragua, and the system, the evaluation system that is working through the website. Maybe in the next week we will open the process, and every person can register free of cost. And we will talk to you about this a little more in detail on the 31st of October. Our perspectives are very good. The last year we had 12 scholarships for this country, and this year we have a quota of 15 scholarships, and we are thinking about slowly increasing these numbers year by year. And we hope to be able to cooperate with Mechnikov, so maybe there could be 15 scholarships for Nicaragua and 5 for Mechnikov. That would be ideal, I think. Thank you very much. Effectively, we dream of the same dream, and I think everybody is very impressed. Thank you, Sofia, for your presentation. Everybody is very enthusiastic about these opportunities for further training that is offered through this program. I think that it is very important that students also learn the Russian language and get the chance to do so in a first year of preparation in Russia. Otherwise, it would be very complicated to study in Russia. So thank you very much, Sofia. Thank you, and an applause for the agency and for Sofia. We also have our representative of Bionika. She's working in Mechnikov and has been doing so for over a year. And she will tell us about her work, how she's working and what she's working at. As far as I understand, you're working in quality control area. Can you tell us a little bit about the destiny and the result of the students that have been studying in Russia and those that have come back already? Good afternoon. Good afternoon. I am a graduate student of UNAM Managua, and in 2018 I started with the Bionika project. We were 8 candidates were selected, and 3 of us were given the opportunity to take an internship in the Mechnikov Institute, and I feel privileged because they told me to work in the management of a quality management area, and we did the work of inspector, so to say, and this helped my professional training a lot because I learned more on the ground as to these aspects. In 2019, after the internship, I was hired as a quality inspector, and it was very moving for me because I had just graduated from university, and it was a great opportunity for me that Mechnikov gave me and helped to develop my professional career in chemistry. I started a new life with new challenges, and we assumed this with responsibility. We had many internal and external trainings. We learned a lot about norms as we are quality managers. And 1.5 years later, I was assigned as coordinator for inspectors, and this was very fruitful for my professional career, as well as enthusiastic, very emotional for me. So I learned even more. I received more trainings. It was much more demanding, but we took all these challenges and overcame every obstacle. So all this has been really wonderful for my professional career. Pharmaceutical chemistry is not a common industry, it's something very special, and we had the doors opened to a new world to us. So I feel very fortunate to belong to the Mechnikov Institute. My colleagues, we are 11 in Bionika in total, and we all have improved and developed our professional career. Some have become specialists. In my case, I started as an inspector, now I'm coordinator of inspectors. I am working in a very active area, involved in productive processes of this institute. Since we started the manufacturing, filling and labeling of our products, we needed lots of knowledge and very special knowledge to be able to work there. We as inspectors have the responsibility to surveil and control all processes and to look out for or guarantee that they are subject to the and fulfill all regulations. So this is an opportunity that I thank my university, UNAM Managua, for, and also the Mechnikov Institute for the opportunity they have given us to strengthen our professional training and develop our careers. Thank you very much. This was a very strong applause for our star leader. We have 11 ambassadors of our projects. One is working in the branch of St. Petersburg. His name is Mario. He is sick, unfortunately, so he couldn't come today. We wish him well. And we can say that these young people are working very well with us. They are helping us a lot. All the young people that come to make their internships, and many of them become employees later. Please stand up so everybody can see you. Thank you very much. We are also online in contact with Pilar Hernandez, the Director of Clinical Studies, the Department for Clinical Studies of the National Regulatory Agency for El Salvador. Can you hear us? Maria, we cannot see you, but we can hear you. Maria, we have a question to you. Can you please tell us about your experience of cooperation with university? How is this interaction model playing out in your country between your agency and university? Now we can see you. I will now repeat my question. We would like to know how the cooperation with university is playing out, and also between universities and employers. Yes, I hope we have a cooperation. On the one hand, with the regulatory agency for industry, and on the other hand, as the National Directorate for Drugs, we have agreements with universities with different programs for students that are close to being graduates or that have already graduated. And we offer them, or we organize internships, and we have a scholarship program in our universities. For example, in the major of biometrics, they have to fulfill 50 social hours. Some of them could be, they are 500 hours, so 250 are of their free choice, and 250 have to be developed in a hospital, and another 100 hours in the industry. So through these agreements, we are giving them other possibilities to the academia, to the universities, not only as to the industry, but also the regulatory agency, because it is very important that we have specialized human resources and well-trained human resources, and also the industry needs these knowledgeable people. So when they come to us, we talk to them about what area they can be participating in. For example, registration, pharmacovigilance, clinical and research. So we offer opportunities, lots of opportunities. As to the regulation of the industry, through the Ministry of the Economy, the training platform, the industry requests specific topics to our ministry, and we offer these. And apart from teaching and explaining some bureaucratic steps or administrative procedures, we can also train them on what the most frequent observations are, how they can repair them or correct them, and then this will help to make a much faster review. And as I mentioned before, it is very important. This axis between industry and academy is very important, and all these hours of practical experiences have been very fruitful because students learn more about the work environment. They participate in courses offered by the companies, because sometimes in the universities they don't learn things so very concretely or practically, and when they go into the world of the industry, they can see things in practice on the ground. As I have mentioned before, our national drugs director, who is the coordinator of the regulatory agencies in Central America, with the support of UNAN Managua, has been looking for training for regulatory agencies, so we can transmit these informations, all this knowledge, to the different agencies in the region, and this will help us to grow as a region. We will broaden our vision. So more or less, this is how the integration is taking place between these three pillars. If you allow me, I would like to congratulate you on this initiative, this Bionica initiative. I will tell you a little bit about my experience. I am a biologist, and sometimes people ask, what are you doing in a regulatory agency? They always believe they have to be doctors or pharmaceutical engineers, chemical engineers. And in fact, one of my bosses in the past told me during lunchtime, as I was eating a salad, please tell me the names of all the species you have on your plate. But I told them, I'm sorry, that was not what I learned. And he was surprised and told me, but you're a biologist, so you should know. But then I told him, no, I studied more about genetics. And then it was very helpful for me to hear about this project, because to communicate all this knowledge, to teach this knowledge, and to broaden the framework of all these scientific subjects and what they can contribute. And often we don't have a correct vision of what a major can offer in the end. So I thank you a lot, and I have already seen lots of fruits. I have heard about the fruits and positive results of this Bionica project, and I would like to thank you again for that. Maria, thank you very much. We are very happy that this project has achieved so much fame that even in El Salvador they know about our work, and it was very interesting to hear about your experience, specifically about the obligation to make internship in social areas for your students, and we might think about some kinds of cooperation. We are also cooperating with a Mexican agency, and we will have another scientific meeting with them. So I hope that and I suppose that in the future we will review our cooperation between us and you. Thank you very much. This is basically all. I thank all the participants of this session for your participation. I would like to tell you something. We have a slogan in our vaccine institute in St. Petersburg that is, the potential is in our human resources. I would like to close this session now. Thanks to all of our experts. And with Sarah, we will start our new Bionica session on the 1st of November. Thank you very much to all of you.